like the peroxicum treated one. Here is the results with relation to the second graph table of the antioxidant enzymes where we studied the catalase, glutathione peroxidase and glutathione reductase, and then you can see the higher level of this enzyme activity in the control one and very reduced one in the arthritis induced, But once the peroxicum treatment was given or the probiotic intervention is given in a arthritis induced, the level of antioxidant enzyme has been increased, suggesting that this treatment is actually favoring the animal to recover the rheumatic arthritis. Next one. Yeah, we actually check the gut microbiota, simplify the microbiological plating, and then we could actually see in the second and fourth week of this duration, the bifidobacteria as well as the lactobacillus count was decreased in a probiotic intervention. However, the pathogenic bacteria, the count was more in the peroxicum treated as well as in the arthritis-induced group. So arthritis group means where the probiotic or the peroxicum treatment was not given. What does this suggest? That when we use the pathogenic bacteria, this result actually clearly shows that the probiotic bacteria, besides their own increase in the antioxidant enzymes where we study the catalase, glutathione peroxidase and glutathione. But with reference to the gut microflora is concerned, it is actually affecting the gut microflora. And the peroxicum treatment was probiotic intervention is given in a arthritis induced, the level of I suggest that this treatment is actually favoring the animals to recover. So after having the basic the culture taxonomic identification of we actually check the gut microbiota, we use this culture to develop the product. The second and we actually use the product higher than the iron, but lesser where it's square. This treatment was done. But we suggest that and we develop this result clearly shows that the probiotic bacteria, besides their own increase the somewhere crosstalk is coming.
Yes.
Okay, thank you. So I was talking about the preparation
Sir, can you
of the bifidobacter.
Can you wind up please quickly
Treatment
because
of
we are already running late, Dr. Prakash.
yeah, a little bit.
Yeah.
Already I am in the product development. Then just I kept characteristics of the product. Okay, so this is what the protocol we follow. Next one. Yeah, so we developed this product and the detail of this products are described here along with its basic probiotic properties and then the preclinical trial results and how this product actually differs. Those health benefits are described here. Next one. Yeah, so this is actually the proximate analysis. This is one of prerequisite whenever we develop any product, how this differs and what are actually the other elements we can actually get. So that information is described here. We have actually followed the standard guidelines, and this was actually in the newspaper also in Indian Express. Okay. I just wanted to emphasize with reference to the safety initial contribution of this Amul, the buffalo milk powder, and then subsequently not much work on the any dairy product in CFTRI. Yeah. So in the context of that one, I just thought I will show this one. So this our benefit of our technology is transferred to this company, Mother's Nature, and Dr. Maria also is actually an outline, and then they are going to start this product. So yes, so they are possible, So she is also there outline on this meeting, and they are going to manufacture this product. So just to summarize, what are the health benefits of this culture if we consume? We will get is described here with reference to the GI, elevating the GI disorder, improving the intestinal microenvironment, anti-allergy, anti-infection, and immunomodulation. In addition to those, the gut microbiome modulation also. Next one. Yeah, so with this, I thank you everyone for giving me this opportunity, and especially giving present this work online mode. So once again, thank you very much. I hope that we get some occasion to interact with the SUSNET delegates in one another day. Thank you.
Thank you very much, sir.
The control is possible from here.
You will check. You just go to strike the we go to. So you just next. We will check.
Okay, that's fine.
Hello.
So first of all, thanks to the organizers for providing me and this opportunity to present in this scientific form on the biotics that are present in the Indian fermented foods, mainly on the fermentation as a key driver for the production of the various biotics, that may be of the probiotics, postbiotics, and the overall the metabiotics, and how they are going to help the human health and the wellness. So as we know, the fermented products are the integral part of our diet and all. So varying across the communities, regions, and with the extensive contributions from these various investigators like Tamam and all. Right now we are having the more than hundreds of the fermented products that have been documented globally. So now with the established science of these fermented dairy products, these fermented products are globally significant, economically valuable, and deeply culturally rooted products. As we know this, when we are going to diverse this probiotic ferment, it's not only the horizontal diversification, it is the transfer of the rich cultural heritage to the next generations. So that is the importance of the fermentation over here, that to the traditional fermentation. Next slide please. So when we are going to see the growth of these traditional fermented milk products, so the fermented milk products or the fermented foods market is growing at the double-digit growth rate. It is accounting the cumulative annual growth rate of more than 14 to 15%, because we know the health and nutrition sales today. So when we are going to talk about the fermentation, so we are going to focus on the other aspects, whether these fermented foods are going to help in the nutritional aspect and also in the health aspects. So as we all know, there is always an increase in the demand for the products that are having the additional benefits like functionality, either of the nutritional, that of the health benefits. So consumer, he always needs the products that are natural and that are minimally processed. So in that, the fermented products category that really shows. So in that fermented foods, of course, the dairy fermented products are also growing rapidly in the fermented functional food category. So we are having the n number of the fermented milk products spanning across the region from north to south and from east to west. So these are the n number of the fermented milk products that are documented from the various regions of the India. Next please. So this is the global map that documented the diverse number of the fermented products globally. So you can see it clearly the Indian position. So in that the India stands out predominantly with the higher color range. So that is exceptional. exceptionally due to the rich microbial and cultural diversity that is associated with India, that is influenced by the various agroclimatic zones and the ethnic tradition, and also the ITKs that are associated with these fermented milk products. So among these, around 600 plus of the documented fermented products, India stands first with more than hundreds of the products that are listed. In that, fermented dairy products stand first. So, and the overall what we can get out of these slides means the fermentation is the key driver that transforms the food matrix into the valuable components through the microbial metabolism and the biochemical modifications that are going to be brought by the microorganisms. So earlier we thought the fermentation is like a traditional practice to preserve the foods, but nowadays this fermentation has explored as the power for the production of the various bioactive molecules, starting from the bacteria to the bioactive peptides and their metabolites. So now this fermentation is having very important role in various concepts like science, in various scientific and the industrial aspects. So fermentation, so traditionally it was just like a food preservation methodology, but this fermentation is nowadays explored as the fermentation is now explored as the tool to produce the food additives, functional metabolites, and it is going to help in the health sector or in the health concept. It is going to improve the nutritive value of the food products, besides aiding in the health aspects, like it is going to inhibit several mode of mechanism it is having in order to promote health. So now it is from the fermentation to the health and functional applications. Now it is expanding to the to the level of the precision nutrition. So that is widely discussed in the keynote address also. So like the precision nutrition means any fermented food, can we use that fermented food for the specific health application. So it is very clear the fermentation is nowadays is widely used it is widely used as the commercial powerhouse for the production of the n number of the bioactive components. From the tradition, it has been transferred to the global from globalization. Next slide. So this fermentation is nothing but harnessing double power of the beneficial microbes, because fermentation will happen only with the microorganisms, and besides promoting the growth of these microorganisms, the food matrix also supports the production of the various bioactive components. So earlier we had a few, means with the less culturable techniques, we had few number of the beneficial bacteria that were documented, but nowadays this list is expanding with the identification of the new types of the beneficial bacteria that play a very important role in the fermentation. So meanwhile, this the growth of this beneficial bacteria, they are also production of the various bioactive components in the food matrix. So overall, the bioactives like the bacteria and their derived and their metabolites like bioactives that are going to promote the health and wellness of the human beings. So before for the better understanding of the nature of these fermented foods, how they will be able to produce the bioactive components, one has to understand what are the microbiological aspects that are associated with the fermentation, which kind of the major flora of the microorganisms that are involved in this fermentation, what is their metabolism, understanding the chemistry of the metabolites production, and also how these metabolites that are produced out of the bacteria are going to promote the health benefits. And then whether we will be able to optimize, whether we will be able to take this traditional knowledge into commercial application, whether we will be able to control all these conditions to get the product that is the same, that will be as same as the traditional product. So, and also when we are going to commercialize this, whether the scalability is possible with all this knowledge, whether do we have the market potential for this kind of the regulatory aspects are very much should be taken care while we are going to commercialize or promote these fermented food products. Next slide please. So how we will be able to address all these things? So we are having nowadays the best advanced tools to understand the culturomics part. Means with the recent advanced culturomics technologies, we will be able to culture the bacteria that are associated with the traditional food fermentation. Of course, if not, because the metagenomics will be there to support what kind of the bacteria that are present in these traditional fermentations and that are present widely in these fermented milk products. and the correlation of this correlation of the data, whether the microbes that are present in the fermented food matrix, really they will be able to produce the metabolites that are present in the food matrix. So, and in addition, the advanced tools like the proteomics, transcriptomics, that can be taken care to understand in detail the fermentation process in these fermented foods. So that are going to, these integrated approaches are going to support us, and they are going to get the better understanding of the traditional fermentation. And so till now I was speaking only the fermentation that is happening in the food matrix, that was promoting the growth of the beneficial bacteria and producing the bioactive. And what about the endogenous fermentation or endogenous application of this fermented food product? Of course, in the exogenous fermentation or the exogenous environment, we will be able to get the beneficial microbial content in that product, and there will be the generation of the bioactive component, and also there will be the possibility of the detoxification of the anti-nutritional components that are present in the food matrix. But at the same time, this food matrix is going to support the bioactives or the bacteria to transit in intestinal condition, and this food matrix is also going to support the endogenous fermentation, the production of the various metabolites like short chain fatty acids endogenously in the gut system, that are going to add again the benefits to the health. So it's not only the fermentation that is happening outside the food matrix, it is also like the endogenous fermentation that is going to benefit the health of the human beings. Next. So, as I told, the fermentation, the food matrix, when it is fermented, it is also going to support the growth of the beneficial bacteria. Several group of the beneficial bacteria have been widely isolated from these fermented milk products, and we are having the regulatory clearances for this category of the beneficial microorganisms under the category of the probiotics. So, it is better to isolate the probiotics that can be able to adapt in the respective food matrix. Next slide. So it is very well known, the probiotics are having the multifaceted health benefits, starting from the pathogen inhibition to the immunomodulation. Next slide. So these are the various, the profile of the beneficial microorganisms that have been reported from India, from various investigators. So from this it is very clear, the traditional fermentation is going to give n number of the beneficial bacteria that can be explored either in the form of the probiotics or the beneficial microorganisms. Starting from north to south, from east to west, we are having the n number of the fermented products and their beneficial microorganisms. With the advanced culturomics technologies, metagenomics, they are exploring the science of the microbiota that are associated with the food fermentation. So fermentation. So after the probiotics, is there any life? So even the dead components of the bacteria are beneficial. That is under the postbiotics. Of course, the fermentation, it is able to produce the certain metabolites that can be taken under the umbrella of the postbiotics. Internationally also the recognition is coming for the postbiotic components. The bacteria after its death and the metabolites that are produced out of the bacteria are also equally capable of producing the same, giving the same health benefits as the live probiotic bacteria. So we all know about these postbiotics, that maybe of the microbial metabolites, inactivated microorganisms, the lysates of the, we know the stages of the bacterial growth. So after the station, there will be the lysis of the cell. Even the DNA of the bacteria is going to matter more with respect to health concept. So overall, the postbiotics are also equally when they are established for the specific health applications. These postbiotics are also going to help us in giving the good health. So now metabiotics. So the bioactive component, that is probiotic, the postbiotic components and the metabiotic, anything derived out of the beneficial bacteria and their metabolism. There may be of any vitamin molecules, there may be of any the important essential amino acids, and the signaling molecules that are produced out of these beneficial bacteria under the category of the metabolites, the low molecular weight components of the beneficial bacteria that are going to improve the health of the human being. So overall, these metabiotics like the consortia of the bioactive molecules after the fermentations that are having the multidisciplinary application that are going to promote even the intestinal health and some metabolites rightly said by some of the speakers like even gamma aminobutyric acid, the tryptophan derivatives, the serotonin the derivatives and all that are going to cross the blood barrier and they are going to action extra intercellularly also. So in the bigger way we are going to get the health benefits out of this fermentation and the related microorganisms. So these are the metabolites that are well studied from the various fermented milk products from the dosa, dahi, buttermilk, lassi, kanji, fermented pickles, fermented beverages, fermented milk products. So these are the very small low molecular weight metabolites, but they are having the important role in promoting the health. So these are the extensive studies related to the fermented foods, how they are going to improve the intestinal health. So we can tell earlier the health is means only we are looking into the gut health of the intestinal health, but the extra intestinal health is also important when we are going to look into the metabolic syndromes like diabetes, obesity and cardiovascular diseases. So they are not going to act only in the intestinal ecosystem, but they are having the significant role to impact the health of the other organ systems also. So again, the fermented foods for the extra intestinal health, they are widely discussed. Their antioxidant activities of the fermented vegetables, fermented pickles, fermented bamboo shoot and all, where the extra intestinal aspects like antioxidative activity, anti-hyperlipidemic activity, and anti-diabetic activities are well explored. So nowadays the meta-analysis studies are also proving the same. So there is a tremendous potential in the fermented food products for the intestinal health. Next. So these are the very important, this is not only from the single studies, from the while analyzing the results of the multi-centered studies, they came to know exactly these fermented milk products are having great role in the prevention of the cardiovascular diseases. So again, the several meta-analysis nowadays we can see where the role of the fermented milk products or the fermented foods are widely recognized globally. So overall, in addition to the intestinal health, nowadays it is widely discussed about the mental health, like the bioactive components or the signaling molecules that are produced out of the fermentations. They will be able to cross the blood-brain barrier and they will be able to modulate the neurotransmission system through endocrine systems. They will be able to reduce the neural inflammations. So like that, the fermented foods are having important or significant role in the extra-intestinal health also. So these are some of the reports from the India where they have extensively studied the microbial diversity of the various fermented milk products. Earlier, the fermentation was thought to be associated with the only few group of the bacteria, but with the metagenomics approach, they were able to decipher more number of the microorganisms that are directly or indirectly involved in the traditional food fermentation. Next slide. So we at India also by inspiring from all these studies, we also done some of the studies in understanding the microbiota and the metabolites of the fermented milk products like dahi from the different regions like north, south, east and west. Next slide. From after integrating all these culturomics and the metagenomics data, we could be able to identify different types of the bacteria that are associated with the key fermentations and the contaminants that were most commonly present in this kind of in the and also the bacteria that are associated with the spoilage of that fermented dahi and by taking care of these other contaminated group of the bacteria and spoilage group of the bacteria, definitely we will be able to under the controlled conditions we will be able to give the good consortia of the bacteria in order to meet the consumer preference of having the traditional dahi at the various regions. So in addition, we have studied the metabolite components of the dahi fermentation also. Interestingly, we could be able to identify very important metabolites that may be of the essential amino acids that may be of the... Only one slide. So like GABA and all that are having very important role in the overall, in the meta health of the human human beings. So that is very clear. So there is a significant transition or transformation from the traditional fermentation to the precision nutrition. So only the science can validate all these claims whether these fermented foods are going to help really in the future. So with all the scientific evidence, nowadays we are expanding the market of the fermented foods. So, okay. So now it is very clear, the fermented foods acts as the multifunctional biotic delivery systems that are going to work both exogenously and endogenously, that are going to work both for the intestinal health and the extra-intestinal health, and these are the futures of the precision nutrition. Thank you very much.
Thank you very much, Dr. Actually, this is a 2-hour lecture. She finished in 20 minutes. Actually, you have at least this needed 4 presentations.
More than 4, sir.
In one. Anyway, thank you very much. What time you are leaving?
Sir, by 4.
This to be
President now. You
trust me.
touch your mobile and
No problem.
Let's go. She is leaving. Here. Come. Come. So now I welcome K.H. Mukta. He is a professor at OB Hero Institute of Agriculture and Veterinary Medicine, and he is associated with Research Center for Global Agroclimatic Agromedicine, Department of Agriculture and Animal Sciences, focusing on forest pathology, myocardial fungi, and tree diseases known for extensive research on forest health, tree mortality, and ecosystem functions. So, thank you very much. Take your own time. We had already given 30 minutes enough.
Thank you very much, Professor Prajapati, for your kind introduction. And I also would like to appreciate Professor Hati and the organization committee to give me such a wonderful to introduce research topics for our project. Maybe not a comprehensive one, but pretty reasonable. Let me start. These are the content of my presentation. Next, please. And to begin with, I would like to introduce a general background about the surface structure of lactic acid bacteria. I will be abbreviating the data afterwards. Can you see here? Maybe it's a kind of picture which is taken from some literature published in 2008. But this is a schematic view of the surface structure of LAB, general structure, and as LAB gram-positive bacteria. It's a single cytoplasmic membrane and thick peptide vital layer like this. Indicating that there are several biopolymers present, such as lipotenoic acid, LTN, and more tenoic acid, and exopolysaccharide abbreviated with EPS, and sortase-dependent protein, these three proteins, and so on. Maybe some of the moonlighting protein could be bound onto cell surface.
You can see on that screen.
Okay, excellent. Thank you. I don't bring pointers. So among them, the peptide glycan and the polysaccharide, the representative polysaccharide, which is included on the bacterial cell surface, and mainly the focus on this presentation. Next, please. So the exapolysaccharide EPS is divided into two groups based on the feature of their chemical structure. One is the homo EPS, like this, which consists of single type of carbohydrates such as glucose and fructose. And like this. And another one is the heterotype EPS, which consists of multiple types of carbohydrates. However, the major components of hetero EPS in lactic acid bacteria are known to be glucose and galactose. And the lactose, those are the major components, and the minor components are gluconate and galactose present. It's known like that. In addition, according to their mode of existence, EPS can be categorized in two groups. One is the capsule polysaccharide, which is covalently bound to the cell surface, and another one is so-called thymine EPS, which is liberated from the bacterial cell surface. Next, please. So the major action of the EPS is supposed to act as a protection against, for example, desiccation and osmotic stress and phage attack and phagocytosis and so on. So on the other hand, some EPS are shown to exhibit beneficial properties for human health, such as antiviral and anti-tumor, prebiotics, antioxidant, and so on. So next, please. So next is peptide glycan. This is a schematic view of chemical structure of peptide glycan. And the glycan chain is composed by the two different types of sugar moieties. One is N-acetyl-glucosamine, and another is N-acetyl-glutamic acid. These glycan chains are bridged with peptides. In LAB, several modifications have been reported. For example, amination in glutamic acid and amination in aspartic acid have been reported. And in addition, the acetylation and deacetylation also have been reported, specifically in the EPS of the lactic acid bacteria. Next, please. So physiologically, the peptide glycan plays a pivotal role in shaping the bacterial cells like this, and it also protects bacterial cells from osmotic stress. In turn, the peptide glycan have been reported to show immunomodulating and host gut barrier stimulating activities. Next, please. Those are the general introduction of the specific structure of the lactic acid bacteria, but from this slide, I would like to introduce two ongoing researches in my laboratory, collaborating with other company and laboratory in my university. First one is about laminose-rich EPS produced by Lacticase bacillus casei strain. So next, please. Okay. Several Lacticase bacillus casei strains were isolated from raw milk of Friesian Holstein cows fed in our region as a producer of laminose containing EPS. You can see this yellow spot. This is showing the presence of the laminose, which is the deoxy sugar of galactose. 6-deoxy sugar of galactose. And because of the highest yield of the crude EPS, we have found 3 strains which produce laminose containing EPS bacteria, but we focus on this virus E2702 strain since its yield of the crude EPS is the highest among them for further analysis. So next, please. We first confirmed presence of EPS-related genes in the strain. As a result of the draft genome sequencing, it turned out there was one potential EPS-related gene cluster. It's very small, but you can see here some genes which is coding the EPS-related genes in one continent. And we also found one compound which is isolated from this one, but it is related to the production, the biosynthesis of DTDP laminols. So it's confirmed that this strain has the potential ability to produce biosynthesis, the exopolysaccharide containing laminols. Next, please. So by using the thoroughly purified EPS produced by virus 2702 strain, several analyses have been performed to estimate the chemical structure of the EPS. And as indicated in the previous slide, the EPS was estimated to be consist of glucose, galactose, and laminols. So from the results of proton NMR, it was confirmed the EPS contains one alpha alumeric hexose and one beta alumeric hexose and 4 alpha laminose residues. Furthermore, we could know the 6 hydroxy group is grouped in glucose and 3 hydroxy group in galactose and 2 and 3 hydroxy groups in the laminose were turned out to form the glycosidic bond. It's related to the formation of the glycosidic bond by this methylation. Next, please. By using the partial hydrolyzate of the EPS, we analyzed them by using LC mass analysis. And as a result, we could know the presence of oligosaccharides, which consisted of 4 laminose residues and 2 hexose residues, as shown in here. So the hex means deoxyhexose, so this is the presence indicating the presence of laminose. Hexose indicating the presence of glucose and galactose. So I don't show the result in here, but glycosidases such as laminosidase and glucosidase, galactosidase treatment of these oligosaccharides indicated that the non-reducing end of these oligosaccharides was the laminose residue. So next, to confirm the order of glucose and galactose residues, their chemical shifts of the alumeric protons were calculated by computer simulation. And as a result, it was suggested that the reducing end sugar was likely to be glucose, since this calculated alumeric proton chemical shift value is smaller than this one. This is indicating the validation. So, and it's also the same, but I don't show the data, but the NMR analysis is also performed by using the chemically synthesized into saccharides. So, disaccharides. So, and it supports, and the data also support the hex disaccharide residue is most probably this galactose glucose residue. So we could know that the hex, double hex residues are galactose glucose. Okay, next please. So to summarize, we reach to this chemical structure of the EPS. And this is so the EPS produced by this strain was the polymer of this neutral linear repeating unit, which consists of 4 laminose residues at the non-reducing end and following beta galactose and alpha glucose with such glycosidic bonds. To determine thorough chemical structure of the EPS, glycosidic binding mode of laminose should be clarified in the future. So we haven't determined which bond is for this bonding, but now we are preparing the oligosaccharides, which is more smaller one, and then we are going to subject this oligosaccharide for NMR analysis. Okay, next please. Next, we integrated the health beneficial function of the EPS especially focusing on the host transpire function. Since this strain is expected to use as a starter of fermented dairy products, so I skip the detail, but by co-cultivation of the system of the CACO2 and HT29 cells, it's known to mimic the human gut exterior in the in vitro system. So we seeded both cells in an appropriate ratio. It was 92:1 to mimic the small intestine onto transwell. And we co-cultivate these two cells, and then we investigated the influence of addition of the EPS and its partial hydrolysis by measuring the TEER and expression levels of the gene and tight junction related genes by real-time RT-PCR using primacies as indicated in here. So next slide, please. Consequently, we found the significant increase of TER here. It's very small, but when EPS was added in this range of the concentrations. Furthermore, mucin-2, mucin-3 gene expression level was also significantly upregulated by the addition of EPS and its partial hydrolysis. And the expression level of human obludin and protein 1 is also upregulated in the similar level of I3PA, which is used as a positive control. So we can see the upregulation of this mucin and tight junction related gene. However, we couldn't figure out those dependency of these expression levels in terms of the concentration of the EPS, so this should be clarified in the future experiment. This is currently ongoing. So next, please. So to summarize this first part, Lactic Casei-Bacillus casei, YRC2703 strain, which is isolated by Yotsuba company, which is the biggest one in the Hokkaido area, where I'm coming from. So it produces a Lambros-rich hetero-EPS consisting of several linear repeating units as indicated here. So to determine solo chemical structure, further experiments should be required, but it's ongoing. And Lambros-rich EPS could enhance expression of mucin and tight junction related genes in vitro. So to clarify its biological benefits when it's applied for preparation of fermented dairy products, further investigation is also required. So next, please. So the second topic is about study on the peptide glycans of several Lactobacillus species. Next, please. It's also the busy slide, but as well known, lactic acid bacteria play also a significant role to maintain healthy status in uterus, because organic acid secreted by them suppress growth and domination of the harmful bacteria such as Escherichia coli and Staphylococcus aureus and Tuberculous pyogenes and some other harmful bacteria. So once dysbiosis happens, such harmful bacteria become major bacteria, and their secondary metabolites such as polysaccharides, LPS, and pyrrolysine produced by Tuberculous. And moreover, the peptide glycan cells also trigger a significant pro-inflammatory response, as meiotic arrest and the inflammatory response, and decreasing estradiol production and so on. So in uterus, resulting in the symptom of endometritis. So next, please. So this is the left one. This is a schematic picture indicating how pro-inflammatory response is evoked by the stimulation of peptide glycan. It's assumed that the peptide glycan binding induced that the peptide glycan binding could induce the heterodimerization of TLR1 and TLR2 that leads this message transfer into the nucleus through such signal pathways. So according to this scheme, peptide glycan stimulation seems to evoke upregulation of pro-inflammatory cytokines such as TNF, IL-1 beta, and IL-6. So in this study, chemically synthesized TLR2-1 agonist PAM3CSK4. This one was used as a positive control. So next, please. So by the way, again, the peptide glycan chemical structure is like this. So, I return to the comparison of the chemical structure of the peptide glycan. So the peptide glycan of harmful bacteria evokes severe pro-inflammation response, but why peptide glycan of lactic acid bacteria doesn't? Most probably some reason may lie in the difference of their chemical structure. So this is the reason why. So this you can see here the chemical structure of the peptide glycan of Staphylococcus aureus, and we can see the clear difference which is surrounded by the dotted red lines. So these differences can be found in the modifications such as acetylation and deacetylation in the glycan chain, and the amination in the amino acid group which forms cross-linking bridge to peptide glycan chain. Next, please. So we first purified the peptide glycan from lactic acid, lactobacillus acidophilus, and Lactobacillus leuceri, and Lacticase-Bacillus rhamnosus. This is in fact LGG obtained from ATCC, and compared them with commercially available peptide glycan purified from Staphylococcus aureus. It's this one, PGN, and this is the peptide glycan from Staphylococcus aureus. And as a result of LC mass analysis of their acid hydrolysis, we found the modified glucanac and murunac residue. In here, you can see the characteristic peaks in PGN from Staphylococcus is like this, but the retention time is apparently different, especially in the peptide glycan from Lactobacillus rhamnosus, LGG. And this peak indicates the presence of murunac in here. But this is a kind of complex of the mixture of the presence of glucanac, acetylated glucanac, and acetylated glucanac, and murunac, or murunac, acetylated murunac, something like that. So since the presence of the acetylation of the glucanac and murunac has been reported, we suspected that maybe the peptide glycan, glucanac and murunac could be acetylated, especially in the LGG derived peptide glycan. So next, please. So we also compare with the composition of amino acid residues. So amino acid composition analysis clearly indicated that Staphylococcus aureus derived peptide glycan base was composed of glutamic acid, glycine, alanine, lysine in this ratio. So it's hard to imagine, but I calculated this, and it's coincident to the chemical structure of the peptide moiety of the generally known peptide glycan of the Staphylococcus aureus. So it indicated that the presence of the 2 or 3 glycine residues in the bridge path, cross-linking bridge path, as reported. So glutamine was not be able to found detected under our experimental conditions. Whereas the peptide glycan lactic acid bacteria also composed of glutamic acid, glycine, alanine, lysine in different molar ratio like this. So it indicated that because of the low content of glycine, we could know that most probably the cross-linking bridge was consist of aspartic acid or aminated aspartic acid in lactic acid bacteria. We couldn't detect the presence of aspartic acid, but you can see here this specific peak is quite close in the vicinity of the peak, which should be detected as and assigned as aspartic acid. So we suspect that this characteristic indicated the presence of aminated aspartic acid, which could be present in the cross-linking bridge part in the lactic peptide glycan lactic acid bacteria. So next, please. Next, we investigated the pro-inflammation activities of the PGNs. From the practical reason, peptide glycan lactobacillus reuteri was omitted. So we just use the peptide glycan from Staphylococcus aureus and Lactobacillus rhamnosus and Lactobacillus acidophilus like this. So just in brief, so it's also the PG slide. So, but just in brief, peptide glycan of Staphylococcus aureus significantly increased expression levels of TNF, CXCL8, IL-1 beta, and prostaglandin insulated genes like this. Yeah, here, this one. Yeah. As similar to PAM3CSK4. However, the peptide glycans of Lactobacillus rhamnosus and Lactobacillus acidophilus didn't. Here. The expression level of TLR2 was significantly upregulated by the addition of all peptide glycans, but on the other hand, the expression level of TLR1 was upregulated significantly by addition of peptide glycans of the Staphylococcus aureus. So interestingly, the effect of the PAM3CSK4 seems to be suppressed partially by the presence of peptide glycans of LAB that might be in a competitive manner. You can see here, this effect of PAM3CSK was reduced under the coexistence of the LAB-derived peptide glycan when compared to this PAM3CSK4 or solo presence. So it's decreased. So okay. So next, please. So in addition, not only the gene expression level, but also we also confirmed that production level of pro-inflammatory metabolite PGE2, prostaglandin E2, which is induced by the PAM3CSK4, was reduced by the presence of peptide glycan of lactic acid bacteria, like this. So next, please. So as in a silico experiment, docking simulation of TLR2-1 complex and its ligands was performed. As a consequence, binding free energies were calculated like this for PAM3CSK. You can see here the number, the smallest number indicating the strong binding. As shown here, PAM3CSK4 could bind to TLR2-1 complex very strongly for both. And its binding was stronger to TLR2 moieties than TLR1 moieties. Here. Similarly, the PGN from of the Staphylococcus aureus could bind to TLR2-1 complex. However, its binding to TLR2 moiety here was about 4 times less than pharmaceutical CSK4. So it's hard to distinguish, but the carbohydrate of PGN can interact mainly to TLR2, and the peptide of peptide glycan can interact to TLR1 here. You can see here, this is a peptide glycan moiety, and this is a peptide moiety, and this is glycan moiety, which interact to PGN1, TLR1, and TLR2 respectively. Next, please. On the other hand, the binding free energy of peptide glycan lactic acid bacteria, the lactic acid LGG's peptide glycan data was used for this, and the binding free energy was 30 times higher than the dose of pharmaceutical CSK4 and the PGN of Staphylococcus aureus in this value. It indicates that the result of biological experiments mainly caused by very weak interaction of peptide glycan of lactic acid bacteria to TLR1. So this binding becomes very weak. So it should be so this weak binding probably interferes the formation of the complex which is required for similar transaction. Similar transfer into the nucleus. And it should be also pointed out that carbohydrate, in this complex, the carbohydrate part was interact with TLR1, and the amino acid residue interact with TLR2. So this is opposite compared to the previous complex model. We don't know why it happened, but maybe some acetylation which occurred in the carbohydrate moiety in the lactic acid bacteria-derived peptide glycan could reduce the binding energy to here, but the cross-linking bridge, which maybe amylated aspartic acid could give relatively good binding energy to the two. This is just speculation. So, as a consequence, we speculated that the difference in peptide moiety is a major determinant for peptide glycan for the formation of TLR2-1 complex. In this context, peptide glycan of lactic acid bacteria, the potential, probably its potential as anti-inflammatory agents in the control of bovine uterus endometritis. So this peptide glycan, it could be also applied in the fermented foods, but further investigation needed, since maybe a similar immunomodulating reaction could be occurred in our small intestine with peptide glycan LAB, but nobody knows, so we need to elucidate this in the future. Okay, next please. So to summarize, Staphylococcus aureus derived peptide glycan and lactic acid bacteria derived peptide glycan differ in the presence or absence of sugar residue modification. The amino acid the sugar residue modifications and the amino acids consisting of the peptide chains and the amino acids forming the cross-linked bridges as well. So the Staphylococcus aureus derived peptide glycan could induce an inflammatory response in bovine endometrial epithelial cells, whereas LAB derived peptide glycan does not. So LAB derived peptide glycan suppressed the inflammatory response of bovine endometrial epithelial cells induced by pharmaceutical CSK4. And finally, it suggested that mainly due to the differences in the minor similitude in the cross-link bridge, Lactobacillus derived peptide glycan interacts very weakly to the TLR1 and it inhibits the formation of the TLR2-1 complex, and thereby it prevents induction of an inflammatory response. Next, please.
Next.
Yeah. So I would like to give my best appreciation to those students and colleagues, and thank you for your kind attention.
We'll take the questions if any after.
This is not indicate this data. I also check the influence of the hexapolyl saccharide against the microbiome on the human feces by using in vitro systems. This is a result of the experiment. Maybe I need to explain this more. We can see some difference in the alpha velocity when you use the feces of the 5 individuals, but it's not for only one individual indicating the significant difference of the alpha velocity. And so it's individual effect. The EPS administration depends on individuals. And this is the kind of comparison of the overall acid concentration. But I couldn't find any of the specific trend by addition of these, for example. So it says that the addition of the 5 microgram of 5 layers of hexapolysaccharide added to the typical fecal microbiota, the influence of the additive addition is based in different form. That's it.
So, thank you. We'll have the next talk. Don't mind, Sarita, can we finish the online? Kanti Kiran is waiting now. Hello, Dr. Kanti?
Yes, sir, I am available.
Let us take your talk because that's what we do not have disturbance and we have only physical talk. So I request Dr. Kanti Kiran is from Navi, Chandigarh.
Sure, sir.
He is waiting since long for his presentation. Please try to finish in 12 to 15 minutes, please. Can you share the presentation?
Yes, sir. Just a moment. Is it seen, sir, now?
No, not now.
Now, sir?
No.
Sir, is it seen?
No. Is there any permission coming from here?
Can you rejoin again?
Yeah, I will try to rejoin, sir.
Yes. परमिशन तो चाहिए ना हमारे देखते Dr. Kanti, are you joining again?
Yes, I am trying to do that. I don't know why it is not coming. Actually, in that share, it is not coming, that presentation. I don't know.
Why share
Have you sent the presentation? We can share from here.
Entire screen.
share
Yeah, yes.
share button is not working.
Sir, I think I will share that, and in the email you can take another presentation.
Okay.
Sir, I will share that in WhatsApp. Can you take other presentation?
Yes, okay.
So we'll start with Dr. Sarita. So we are taking the other speaker. In the meantime, you can send.
Sure, sir.
Okay. So Dr. Sarita Utala is from Maharashtra, Pune, and she is professor of microbiology.
Okay.
Yeah.
Good evening all. So today I am delivering my sessions on the 12th International Conference on Fermented Foods, Health Status and the Social Well-being. And my topic is, next please. My topic is lactic acid bacteria as bioengineers of wellness, opportunities for food security and public health. Next please. So here, why I selected this topic? Because of there are so many problems. The major problems we are facing: rising non-communicable diseases, global food insecurity and malnutrition, as well as the antibiotic resistance crisis. So to overcome these problems, here there is a huge demand for the natural sustainable health solutions. Next please. So here the first point, that is the rising non-communicable diseases. The non-communicable diseases have reached the pandemic proportions, becoming the leading cause of the death and the disability worldwide. And in India, the current scenario is characterized by the ongoing epidemiological shift from the infectious diseases to the chronic conditions, driven by the lifestyle, environmental and the demographic changes. Next please. Here there is also another problems, that is the global food insecurity and the malnutrition. So in that cases, here in 2025, the global scenario is stated that the approximately 673 million people, 8.2% of the population is facing the chronic hunger. And in that cases, The slight decline from the previous year's progress is uneven. Hunger continues to rise in Africa, where 1 in 5 people are affected, and in Western Asia also. Next, please. The another problem that is the malnutrition, and here we are also facing the child undernutrition, obesity, micronutrient deficiencies, as well as here there are so many conflicts that leading the cause of the acute food insecurity, impacting approximately 140 million people in 20 countries. The another is the climate change. This is also the extreme events that push more than the 96 million people into the food crisis in 2024, and the forced displacement. So here in that case, the approximately 95 million displaced people live in food crisis setting with the limited livelihoods. Next, please. The challenging problems, that is the antibiotic resistance crisis. And here we are observing that so many pathogens becomes the antibiotic resistant, they becomes the superbugs. In that cases, we are observing that the fluoroquinolones, this is the highly resistance in the diarrheal bacteria and the typhoid cases. Whereas the another drugs, the carbapenems, in that cases the E. coli and the Klebsiella, the susceptibility down in the 57% as well as the 31 to 35% respectively. Whereas there is a case of the ICU infections, the most of the dangerous, and in that cases the Acinetobacter baumannii, it shows the 91% resistance. Whereas the carbapenem resistance up to the 43%, and whereas the ventilator associated infections, pneumonia, this is the 80%. These are caused by the Acinetobacter, Klebsiella as well as the Pseudomonas. Another study that stated that the gram-negative bacteria that dominates the major causes of the UTI, pneumonia and the sepsis, and the most common superbugs. These are detected as the E. coli, Klebsiella pneumoniae, Pseudomonas aeruginosa, Acinetobacter baumannii and the Staphylococcus aureus. And in the bloodstream infections, in which there is a 72% caused by the drug-resistant gram-negative bacteria and the 10% caused by the fungi. Next, please. So here, to overcome these challenges, there is a demand for the natural and the sustainable health solutions in pathogens, as well as this is also useful for the producing the antimicrobial peptides like bacteriocin, acetic acids, propionic acids that also eliminate the competing microbes. It is also the safety barriers during the fermentation process. It acts as a backsloping and also prevents the proliferations of the dangerous organisms. Next, please. So here lactic acid bacteria organisms, these are also useful for the textures as well as the flavors of the product. Next, please. So here this is the mechanisms in that. Here we can use the probiotic as a therapeutic purpose, metabolic engineering, functional food design and the precision nutrition. So these smart probiotics, engineered strains we can use in the functional probiotics, conventional probiotics and next, please. Here there is a use of the metabolic engineering and the synthetic biology for the precision nutrition. There is a use of the CRISPR technology as well as the functional food designs and the precision nutrition. In that, the LAB are critical for shifting from the general nutrition to the precision nutrition, where the foods tailored to individual health needs. Next, please. So, in that cases, here the functional foods, these are the conventional nutraceuticals. In that, there is a use of the probiotics as well as the prebiotics. And due to this here, we can also cover in the host, there is epithelial barrier, innate immunity, adaptive immunity. Due to this, there is a modulation of the immune response. We can also use nutrient digestions, vitamin productions, short chain fatty acid productions. So here these are the productions of the metabolites through the LAB. And here also there is a microbial host interactions. In that, dietary nutrients, dietary fiber and the phytochemicals. These are the part of the precision nutrition. Next, please. There is also another concept of the FOSHU. FOSHU is the food for the specified health uses. In that, there is a use of the specific components, prebiotics, and we can control the health conditions, including the blood pressures or the blood cholesterol. Next, please. there is a diseases and there is the functional foods and the ingredients which are used to control the various diseases with the help of this functional food. Next please. So here we can prepare these novel functional foods with the help of the bioactive compounds which are present in the lactic acid bacteria, cell-free supernatant, as well as in the various vegetables or the fruits. With the help of this, here we can also use the probiotics and prepare the functional foods. So this is the novel functional foods. Next please. Here the natural sources, in that there is the use of the various bioactive compounds incorporated into the foods and the probiotics. Next please. So with the help of these bioactive compounds, these are the organic acids, bacteriocin, exopolysaccharides, vitamins and the cofactors, bacteriopeptides, AC inhibitors and the GABA productions. All these bioactive compounds, these are present in the cell-free supernatant of the lactobacillus cultures, and this plays an important role for the health benefits. Next please. Next please. Yeah. Next please. So here the work in that near about 190 bioactive compounds. Among these 190 compounds, we found the 97 bioactive compounds through the metabolic profiles. To metabolomic profiles, this suggested that the presence of these phenolic compounds, organic acids, short chain fatty acids, and the hydroxy fatty acids, phenolic acids. Next please. Next please. Next please. So in that there are the various vitamins, these are present. Next please. This is the LCMS profile of the bioactive compounds. Next please. So here through these mechanisms, here we can understand the various bioactive compounds which are present in the lactobacillus cell-free supernatant. This plays an important role, and many more references, they suggested that these bioactive compounds plays an important role to support the gut epithelial energy, hemostasis, suppressant, as well as enhance the host metabolic flexibility. Next please. Here another short chain fatty acids or the hydroxy acid pathways, these are indicates that the anti-inflammatory signaling, they improve the gut barrier integrity, antifungal as well as the antimicrobial protection. Next please. The another tryptophan catabolisms, the endon signaling pathways. This also indicates that the health benefits, that is the maintenance of the gut mucosal hemostasis, protection against the IBD, and the regulations of the host microbe immune prostom. Next please. And that there is a physiological relevance of these phenolic biotransformations pathway, antioxidant defense, anti-inflammatory activity, protection against the foodborne and the gut pathogens. Next please. The another fatty acids and the hydroxy fatty acid pathways, that physiological relevance indicates the anti-inflammatory lipid signal balance. Next please. The amino acids, these are present in the lactobacillus cell-free supernatant, that indicates the different activity like stress and the anxiety modulation, enhance intestinal repair mechanisms, neuroimmune communications, as well as there is the presence of the neuroactive metabolite pathways. This plays an important role in our health benefits. Next please. The next one is the peptide and the DKPs or the redox molecules, which are also indicates the oxidative stress protections, immune priming, pathogen surveillance. Next please. The EPS, presence of these EPS carbohydrate signaling pathways, that indicates the immunomodulations, antiviral protections, and the prebiotic-like effects. Next please. So here we can prepare the various functional foods with the help of these probiotic organisms, because we know that in the fruits or the vegetables there are so many phytochemicals are also present. So with the help of this LAB and the prebiotics, we can prepare the functional foods. And here in our lab, we prepare the various juices as well as the beverages. So here there is examples of the guava juice, in that we add the probiotic organisms and prepare the probiotic juice of guava. Next please. Next please. So next please. Next please. So in that there is a presence of so many phytochemical compounds. We analyzed with the qualitative methods as well as the quantitative methods, and we found there are approximately 97 bioactive compounds are present. Next please. We also studied the control as well as the fermented the juice of the guava, and we found that the not only the bioactive compounds enhanced, but here there is a biotransformation we also observed, and more than 10 another bioactive molecules we found. Next please. Next please. Next please. It is also prominent in the antimicrobial activity. Next please. It also shows the significant antioxidant activity. Next please. It is also useful in the case of the anti-diabetic activity. We carried out this activity by the alpha amylase inhibition methods. Next please. We also carried, this is the SMS profile of this juice. Next please. Next please. Next. The another, so here this fermented juice, which is also claims for the anti-diabetic activity, antioxidant activity and the anti-cancer activity. The another juice that is here, you can observe that the dragon fruit juice we prepared. Next. Next. Similarly, we also prepared the, next please, the pumpkin juice. Next please. Next. Carrot juice. Then there is also the fermented finger millets beverage we prepared. Next please. Next. We carried out the 9 hedosconic sensory test. Next please. We also carried out the another one biotransformations of the beetroot and its bioactive compound for the formulations of the nutritional gummies. And with the help of these probiotic organisms, we prepared the gummies. Next please. Here, the Lactobacillus plantarum species and the fermented species we used. We carried out the LC.
I want to present everything at once. These are 4-5 presentations could have made. Focus on one product and go to the system. Anyway, please come.
Yeah. Next, please. These are the compounds. Here we indicate that the beta-sanins, the beta-xanthines and the total polyphenols, antioxidant activity and vitamin isomers. So here the concentrations of these increased, and also we'll find out the another bioactive compounds. Next please. So these are the listed compounds. These are present in the fermented beetroot gummies. These are absent in the control. Next please. These are the listed compounds. Next please. So these are the gummies prepared. Next please. Next. So this lactic acid bacteria, these are the very useful. Prepare the lactic acid bacteria based functional fermented foods is a practical and a sustainable public health strategy that offers the significant nutritional and the preventive health gains with the minimal economic burdens. So we can say that in the this scenario or this era, the lactic acid bacteria plays an important role in the functional foods. These are useful for the well-being, as well as here we can prepare the foods with the safety and the security. Thank you so much.
Actually, we are asking both.
Yes, yes.
No, but you have to go very hurriedly, na? That is the issue. Anyway,
Slide cell.
now Dr. Kanti Kiran is there on the.
Huh?
Okay, we'll start with
Mr. Rajesh.
Mr. Rajendra. Dr. Kanti just
Can
made
you download
this
what
downloaded. We will finish Dr. Mishra Ji talk.
Mr. Rajesh, follow.
So Dr. Mishra is from NEHU and he is welcome to all of you. He will now talk on his very important topic, antibiotic associated diarrhea.
So, respected Chairman Sir and Protea and all my colleagues and students, good afternoon everyone. Now I am going to present my topic, that is effectiveness of lactic acid bacteria and yeast in managing antibiotic associated diarrhea. Next. Actually, antibiotic associated diarrhea is a frequent adverse effect of antibiotic therapy and affecting 5 to 39% of patients depending on age, population and the antibiotic used. Older adults over 65 years are at higher risk, particularly when treated with broad spectrum agents such as clindamycin, cephalosporin, and many other antibiotics. Although no consensus exists on the highest risk of antibiotics, any agent can disrupt the colonic microbiota and induce diarrhea. Antibiotic-associated diarrhea may develop up to 2 to 3 weeks after therapy ends. It is especially problematic in hospitalized, frail patients, often resisting premature antibiotic withdrawals, prolonged hospital stays, increased cost and high risk of infection and mortality. Next please. So, why antibiotic associated diarrhea occurs and how probiotics may prevent it? Antibiotic actually disrupt all colonic microflora, change the carbohydrate metabolism and increase diarrhea, cost and hospital stay, especially with motility enhancing drugs. Probiotics including Lactobacillus, Bifidobacterium and Saccharomyces etc. may reduce antibiotic associated diarrhea by maintaining gut microflora and inhibiting pathogenic bacteria. Next. So we have isolated many lactic acid bacteria. Out of that, 2 lactic acid bacteria and 1 yeast species we have identified, and we have checked their probiotic attributes. We found that highest starch hydrolysis activity, glucomylase activity, alpha amylase activity, beta amylase activity was attributed by Lactobacillus fermentum, while Lactobacillus plantarum showed maximum dextrinase activity. Probiotic attributes of selected isolates were performed, where Lactobacillus plantarum showed highest pH tolerance, cell autoaggregation and organic acid, lactic acid, butyric acid and production of vitamin B2 production, whereas Lactobacillus fermentum showed maximum bile tolerance, antioxidant potential, vitamin B12 production, cholesterol assimilation, EPS production, cell surface hydrophily and vitamin B9 production, whereas Saccharomyces cerevisiae showed maximum pH tolerance, antioxidative activity, vitamin B12 production, cholesterol assimilation, acetic acid production, EPS production, butyric acid production, adhesion capacity and vitamin B2 production and vitamin B9 production. Next. This is the overall view of the experimental design. We have taken the test groups. Each test group contains 6 fish-star rats. 1 ml of each sample was fed twice daily till 14 days. Test group A1, that was functional fermented rice beverage. It was fed to in 1 ml dose. Test group 2, A2, Lactobacillus plantarum and Lactobacillus fermentum was given into 1:1 ratio. And test group A3 was Saccharomyces cerevisiae suspended in PBS and diluted accordingly to adjust the cell counts. 8 × 10⁻⁷ log CFU per gram. Test group normal control fed with normal diet without any other treatments. Test group disease control antibiotic mixture for inducing diarrhea. And test group standard, this was fed with loperamide drug for inhibiting diarrhea. Next. And we have checked the diarrhea assessment on the basis of body weight, fecal consistency, fecal water content. Next. And detection of fecal starch and fatty acids, we observe acetate, lactate and propionate. Next. On hematological analysis, we see the red RBC, hemoglobin, hematocrit, mean cell hemoglobins concentration, mean cell corpuscular volume, mean corpuscle and hemoglobin concentration. Next. We have taken the pooled fecal sample analysis for lactobacilli cell, enterococci cell counts, yeast cell counts and coliform counts. Next. And we have developed the defined starter culture. On the basis of this starter culture, we have developed the fermented rice beverage and we fed in 1 ml dosage for the test group A1 mouse. Next. 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 we have already discussed. Next slide. So total 36 Vista rights were divided into 6 groups, that is normal control, disease control, standard A1, A2 and A3. Normal control was treated with saline for 14 days. Disease control group was administered with antibiotic mixture, and standard group was administered with antibiotic mixture and loperamide, and test A1 group was fed with antibiotic mixture for 14 days and fermented rice beverage was administered twice a day for same time period. Test A2 group was fed with antibiotic mixture for 14 days and the combination of Lactobacillus plantarum with Lactobacillus fermentum was administered twice a day for the same time period. Test A3 group was fed with antibiotic mixture for 14 days and Saccharomyces cerevisiae was administered twice a day for same time period. Next. And we have already discussed this in earlier slides. Next. So we have checked the fecal consistency after feeding all these groups. So out of all the groups except normal control, the functional fermented rice beverage was comparatively least affected by antibiotic associated diarrhea with a score of 1.183 plus minus 0.166 on day 5 and on day 10 and completely subsidizing the diarrhea on day 14 with formed stool with a fecal consistency of score 1. On the last day of study, the test group A1 and standard showed dry and normal consistency of feces with well maintained shape and brown color with fecal consistency, score of 1, presuming to derive the antibiotic associated diarrhea was resolved. Next. We have seen the fecal water content. So after day 10, a significant reduction in the fecal water content was observed in the A1, A2 and standard till day 14, while the fecal water content of other groups like normal control, disease control and A3 was not significantly reduced after day 10. Next. Yes. So we have observed the body weight. The body weight of the normal control group showed no changes till 14th day of the study. A slight decrease in the weight was observed in rest of the test groups after 5th and 10th day of the study, which eventually was at par with the body weight of the normal control group after 14th day of the study. Next. We have seen the microbial analysis of the pooled fecal samples. Although a cell reduction was observed on day 5, but comparatively amongst all the test groups, group A1 showed higher cell count on day 5 for lactobacilli, enterococci and yeast. Next. We have observed the hematological analysis. So, based on the above hematological results, our sample A1, that is fed with fermented rice beverage, can be considered predominant over the test groups in the study for resolving the deleterious effects of antibiotic associated diarrhea. Next. We can see here the histopathology analysis of colon. Test group A1 not shown any abnormal changes that indicate it could work well as anti-diarrheal drug. Next. We have seen the histopathological analysis of the liver. We can see here in the picture test A1 group, which was fed with fermented rice beverage, did not reveal any specific lesions and is at par with the normal control test groups, indicating that it could work well as anti-diarrheal drug. Next. We have observed the cecal sartan fatty acid quantification from antibiotic associated diarrhea animal models. The cecal concentrations of lactate, acetate and propionate were significantly higher in the rats of test group A1 fed with fermented rice beverage as compared to rest of the groups. There was no presence of acetate in the test groups normal control, disease control and standard. Next. Next. So, in conclusion, we can say the treatment with fermented rice beverage, that is sample A1, could contribute into the maintenance of structure of the right mucosal microbiota in comparison with the normal group and disease control group. Moreover, it had protective effects on the gut structure and well maintenance of the body weight, fecal water content, fecal consistency, score similar to normal control group. Results from the histopathological analysis, that is colon and liver, hematological analysis and short chain fatty acid production from the rat fecal contents proves it to be an efficient drug in preventing the antibiotic associated diarrhea. Next. So this was our team. This project was funded by DBT Government of India. From my side, I was the principal investigator, and from Kamdon University, Dr. Subrata Hati was the principal investigator. So, thank you.
So now we have a last speaker, Dr. Kanti Kiran from Navi, Chandigarh.
Sir, can I share the slides?
Yes, you can.
If you can do it, well, good.
Is it seen, sir?
Your screen is seen.
Yes.
Yes.
Yeah.
Yes.
Yes, sir.
Yes.
Please continue.
May I continue, sir?
Yeah, please distribute your.
Yeah, very good evening to the gut microbiome. So our healthy gut is harbored by countless number of microbes in the small and large intestine, which totally called as gut microbiota. And we all know, and it is vastly accepted, that microbial changes are associated with various kinds of diseases. It can be communicable or it can be non-communicable diseases. And manipulation of the microbial composition by either diet, fecal microbial transplantation, or utilizing prebiotics, probiotics, and symbiotics, postbiotics, and certain drugs, we can achieve the intestinal homeostasis, wherein the tight junction proteins can be restored, and the metabolites, harmful metabolites going from the intestine into the circulation to various peripheral organs, and the central organ can be controlled, and hence we can control the diseases. So among the various approaches, my talk will be focusing on developing probiotics, prebiotics, and symbiotics with reference to obesity. So obesity is one of the non-communicable diseases where there is a dysbiosis in the gut bacteria which communicates with the adipose tissue, leading to harmful metabolites accumulate in the gut, and in the adipose tissue causes adipose inflammation and obesity. So we have been, last 15 years, we have been working with various millets and dietary bioactives from these millets, and we have shown through various publications that finger millet and kodo millet especially are very potent in preventing harmful changes that is caused by high fat diet and control the obesity. And most of these changes are associated with the changes in the healthy gut bacteria, which otherwise dysbiose with high fat diet. And this millet bran and millet bioactive supplementation along with the high fat diet restores the intestinal microbial composition and promote the health and reduces obesity. So we have recently developed a modified kodo millet bran by chemo and enzymatic methods, which renders this millet bran into a bioactive ingredient and hence can be used as a prebiotic source. So coming to the probiotics, our lab is been working on probiotics since almost 20 years, where we have isolated hundreds of bifidobacterium strains and lactobacillus strains, and through rigorous in vitro screening process following ICMR DBT guidelines as well as international agencies like EFSA, we have found that two bifidobacterium strains, Bifidobacterium longum and Bifidobacterium brevi, Rhamnosus and Plantarum belonging to the lactobacillus were potent in reducing the inflammation. On this panel you could see the electron micrographic images of these bacteriums. And then we have done whole genome sequencing of these 4 bacterial strains, and then we have determined its safety in vivo using male and female animals as per the OECD guidelines, and following acute, subacute and subchronic toxicity studies, and we found that these bacteria are safe as they do not cause any unwanted changes in the organisms, especially in the rats. So following that, we have our aim was to develop a cocktail of bacterium as multi-strain combinations of Bifidobacterium and Lactobacillus strains. So this is an in vitro model where we have shown that intestinal epithelial cells, when treated with palmitic acid and lipopolysaccharides, which are the main culprits in causing obesity, when we challenge onto the Capo2 cells, you see there is a lot of lipid accumulation here, and this bacterial treatment along with palmitic acid and lipopolysaccharide is able to reduce the lipid accumulation. So more green color here indicates more lipids, and less green color indicates less lipid accumulation. So this is the quantification. As you see here, palmitic acid treatment enhances the lipid accumulation, while bacterial treatment along with palmitic acid and lipopolysaccharides prevents this. So similarly, we have shown through, we have also seen the stability of the tight junction proteins. And when this bacterium and along with the lipopolysaccharides and palmitic acid is given, the tight junction proteins are restored. There is no loss in the tight junction in the intestinal border. And we have also determined through fluorescent labeled dextran dye, more fluorescence in the basolateral chamber. In the lower chamber indicates the leaky gut, more which was prevented by the bacterial strains. So we have also done a co-culture study between the Capo2 cells, and the lower layer is having the livers and liver cells, and LPS and palmitic acid and bacteria are given in the upper chamber on the intestinal epithelial cells. Due to prevention of the leaky gut, there is no molecules going into the lower chamber and affecting the hepatocytes. So as a result, the Capo2 cells treated with palmitic acid lipopolysaccharides through the intestinal membrane, lipid accumulation is accumulated in the liver cells, which was prevented when bacteria are treated on the intestinal epithelial cells along with the palmitic acid and lipopolysaccharides. So we have also seen the gene expression in the hepatocytes. The palmitic acid and lipopolysaccharide treatment significantly enhanced the gene expression of fatty acid synthesis, while the treatment with bacterial strains on the upper chamber prevented the lipid accumulation, suppressing the lipogenic gene expression and increasing the insulin resistance and glucose homeostasis genes. So we have also shown that these bacterial strains, when we mixed with the bioactive codomillet bran, stimulated the healthy gut bacteria, and most importantly, it produced, this is in the human fecal batch fermentation studies, and most importantly, butyric acid is found to be enhanced with the fermentation with this bacteria and the codomillet bran. So we have shown that this symbiotic blend combination of these four bacterium and along with the modified codomillet bran, it could prevent the obesity. This is a high-fat diet induced model. These are the various groups: normal diet, high-fat diet, symbiotic with high-fat diet, symbiotic with the standard drug. As you can see here, after the experiment, that animals fed with the high-fat diet showed higher body mass due to fat accumulation, which was confirmed through cutting open the animals as well as through DEXA studies, while the bacterial treatment, symbiotic treatment along with the HFD prevented the body weight gain, which is also evidenced through this DEXA as well as the whole organism visualization. So we have, this is also, we have quantified the weight gain. High-fat diet promoted the weight gain. The saroglitazole, which is a standard drug along with the symbiotic, is able to supplement, suggesting that symbiotic supplement is at par in preventing the weight gain as compared to the standard drug. And we have also seen in the liver, histological assessment of the liver. In the high-fat diet fed animals, the liver is laden with the high-fat globules, which was prevented in the saroglitazole plus high-fat diet as well as in the symbiotic diet fed animals, where the histology looks equal to that of the control animals in these two groups. This is also a visualization of the liver. More red color is more healthy liver. More pale or off-white color indicates most lipid accumulation, which is higher in high-fat diet compared to the symbiotic as well as standard drug treated group. So we have also seen these various lipid profile parameters. As we see here clearly, the cholesterol levels and triglyceride levels are high in the high-fat diet, which was prevented by this symbiotic treatment. We have seen through proteomic analysis, the genes responsible for fatty acid synthesis and absorption is more in the high-fat diet, which was prevented by the treatment with symbiotic group. So this is also, as you can see in the blood glucose homeostasis, insulin resistance is high in case of high fat diet. Symbiotic and saroglitazole treatment in separate groups reduced the glucose, prevented the glucose increase in the serum, suggesting that it has insulin sensitivity. In this scenario, the symbiotic blend is equal to the saroglitazole, which is a standard drug we have used. As you can see here in the histological session, HFP promoted the atrophy, that means increase in the size of adipocytes, which secrete pro-inflammatory adipokines that is responsible for the adipose tissue inflammation and obesity, which was prevented by symbiotic supplementation. So to summarize our study, that high fat diet causes significant damage to the intestinal breast border, which in turn causes hepatotoxicity in the liver as well as atrophy in the adipose tissue. Symbiotic supplementation along with bacterial mix and the 10% of bioactive bran significantly prevented this leaky gut as well as protected the liver and adipose tissue from the hypertrophy. So this, our whole study suggested that in vitro bacterial strains are able to prevent the lipid accumulation, as well as in vivo models we could suggest that the symbiotic blend has anti-obesity by preventing adipose hypertrophy and steatosis in the liver. So I would like to thank the funding sources, especially to the conference organizers, Dr. Subrata Hati, Dr. Prajapati and other Dr. Lopamudra for giving me this opportunity to show our results and discuss our results. And I would like to thank my funding sources, collaborators and everyone. Thank you. And this is the student who has carried out the work.
Thank you. Thank you so much, Dr. Gandhi.
Thank you, sir.
Final picture of closing.
Thank you so much, sir, for this wonderful session. In this session, we are delighted to have Dr. J.B. Prajapati as chairman of this technical session, as well as we are extremely happy that Dr. Subrata Hati is here as a rapporteur. So I would like to request Dr. Chaitanya Chakraborty of this faculty to provide a token of gratitude to our honorable chairman, sir. Now I would like to request Dr. J.B. Prajapati sir to honor our invited lecturer who have come from far away and who have presented beautiful lectures here. So the first name is Dr. Sarita. I would like to invite Dr. Fukuda. Then I would like to invite Dr. B.S. Mishra. B.K. Mishra, sorry. I'm extremely sorry. Thank you so much. So this is the end of this session. But in this room, the next session is going to start from within 5 minutes.
Direct 3 PM come back means before 10 minutes, please.
तो speaker को भी बुला लो, फोन नहीं किया ना भाई, all the speaker please come, we are having one photo session.
Dr. Kanji.
Now,
We will now start technical session 3. So be here and we have tea here. So students are requested to be present here in the hall. Yes.
चाय इधर ही मिलेगा भैया.
Don't go. Please. Tea will be served here. And we are just within 2 minutes starting our technical session 3.
ब्रीदिंग टाइम रखा है नहीं. 120
কোনটা
Hello, Dr. Jogesh Khetra, please come to the seminar hall, first floor. Dr. Jogesh Khetra, we are starting our technical session 3 and the invited speakers for technical session 3 are requested to come to the seminar hall 1 that is in the first floor. For technical session 3, Dr. B.K. Mishra will be the chairman and Dr. Jogesh Khetra will be the rapporteur. So chairman is already with us. Dr. Khetra is requested to come to first floor in the seminar hall so that we can start the session 3. All the invited speakers are requested to come forward. Kaushik sir is here. Krutika Abhayankar? Yes. Dahikar? Oh. Keshav Chandra Mondal? Keshav Chandra Mondal? PM Raju? Sanjeev Kumar? And Dahikar is absent? Dahikar Samadhan? Okay, okay. And Divas Pradhan is there? Okay.
Hello.
हाँ
आ जाओ.
Divas Pradhan is there? Yes, Divas
दहीकर नहीं है डॉक्टर समाधान दहीकर नहीं है
Dahikar and Sanjeev Kumar? Dr. Khetra?
He's coming.
Okay. Slide change करो ना next बोले slide पे change करो आप एक तो देख सकते
कहाँ से बोलना है आपको?
हैं
स्पीकर वहाँ खड़ा होंगे ना?
एक बार सर आने से होता है एक तो सर नंबर है सर
हाँ फोन खोलना होगा वास्ते अलग आ जाओ तो क्या शुरू करवा दे क्या ठीक है ओके हम 5-10 मिनट लगेगा उसका अब 4-10 मिनट
A very warm welcome to all the dignitaries, eminent speakers and dear participants. We now proceed to technical session 3 of the UFSASNetCon 20 session, fermented foods and their functionality. Is very highly relevant in today's context as fermented foods are increasingly recognized for their nutritional, therapeutic and functional attributes, contributing significantly to human health and well-being. It is my privilege to inform you that this technical session will be chaired by Dr. Virendra Kumar Mishra and the rapporteur will be Dr. Yogesh Khetra. So in this session, 5 invited lectures will be delivered and I humbly request all speakers to kindly adhere to the allotted time to ensure smooth conduct of the session and allow sufficient time for discussion. May I now request the Honorable Chairman Sir, Dr. B.K. Mishra to kindly take over the proceedings of technical session 3. Thank you.
So thank you, madam. I welcome all the speakers and students, my colleagues here, those who are present here. So in this session, there are 7 speakers, 2 are absent. So only 5 are present. So one by one, I will request all the speakers to come and present their deliberations. So first speaker of this session is Dr. Divas Pradhan. He is alumni of this institute and currently working as senior scientist at Dairy Microbiology Division, MDRI Karnal. So I will invite Dr. Pradhan, please come and I will request you please complete your speech within 12 to 14 minutes.
Okay, so first of all, thank you to the organizers for giving me an opportunity to present one of the work that we recently carried out in our lab, which is titled Comparative Functional Analysis of Milk Fat Globulin Membrane from Cow, Goat and Human Milk in Urine Gut Model. इसको थोड़ा मूव करो ना, यहाँ से भी कर सकते हो, ऊपर से सर ऊपर तक करना। Next. So, like the gut health is one of the focus of the recent times, and people are trying to modulate the gut microbiome so that we have better health in terms of the gut microbiota, and it is linked with many other diseases and all. So based on this, people are trying to devise probiotics, synbiotics, prebiotics to improve the gut microbiota. So that's the target of the all these things that we are talking about, which are the probiotics, prebiotics and all. So a couple of things that are not really people are not really trying to look into is what is the impact of food molecules, food components on the probiotic transition in the GIT, and as well as what is the effect of the food components itself in the gut health. So milk fat globulin membrane is one of the already recognized, very well recognized molecule, especially milk fat globulin membrane from human milk has been recognized with a lot of health benefits. There are milk fat globulin membrane is made up of, one is the proteins and another one is the phospholipids. So proteins and phospholipids, there are some that has been mentioned here, and it is already known that this contains a lot of these health benefits. So what we basically did was we took the milk fat next. So what we basically did was we collected the milk from cow, goat and human milk, and we isolated the milk fat globulin membrane. So that was one thing, the isolated milk fat globulin membrane, and the milk fat globulin membrane we prepare to hydrolyze it, because in our system when we take whatever we are taking, it is hydrolyzed based on the passage and what are the enzymes that is available. And at the same time we also treated the bacteria. Here we use the bacteria Lactobacillus rhamnosus LGG, which is a classical probiotic. We dip the bacteria in the MFGM hydrolyzate, and we again extracted back the bacteria. So, and then we tested further that what exactly it does in a gut system in both in vitro and in vivo model. Next. So first of all, in order to understand that our MFGM is not toxic to the cells, we ran a toxicity test, and based on that we understood that a level of 500 microgram per ml is a non-toxic and standard dose for testing. And then we So then what we did was, first of all, we tried to look look at whether our MFGM extract that we had, it is interfering with the addition of the bacteria. So here we see that when we are adding MFGM and we are looking at the bacterial addition, it is interfering. That means the MFGM hydrolysate is occupying the sites where the bacteria is interfering. But if the bacteria is already being treated with the MFGM, the addition is increasing to a huge level. So what we probably when we also looked at the conformal level and all, we are postulating that the MFGM is kind of coating the bacteria, which is increasing the addition to a large fold. So at the same time, we also saw the permeability. So we ran the permeability test by using LPS and inflammatory molecule, and we try to see that whether our treatment with MFGM or the bacteria is able to bring down the inflammation. So the most effective was the bacteria that was treated with it, right? And at the same time, we also looked at whether the pathogen exclusion. So whether our treatment of bacteria and MFGM, whether it is able to give a protective competitive displacement effect against pathogens like Salmonella, Typhimurium, and E. coli. And here we could see that the bacteria that is treated with the MFGM was able to give a very high effect in all the protective competitive displacement with both the bacteria, Salmonella and E. coli. And then we looked at the inflammation by inflammatory cytokines and inflammatory pathways like the TLR4, MID88, NF-kappa B pathway. When the LPS was being treated, there was high expression of all the pro-inflammatory pathway markers, and at the same time, when we were looking at the treatment with other kind of treatment, we saw that there was a lot of repression of the pathway. So we saw that the MFGM as well as the MFGM that was the bacteria that was treated with MFGM was bringing out the inflammatory markers. At the same time, there was other stress markers like COX-2 and HS70, which also showed the same kind of pattern. And finally, the final inflammation comes in terms of the pro-inflammatory markers like IL-12, IL-1 beta, and IL-8. And here also we saw that when it was being treated with LPS, lipopolysaccharides, these markers were highly upregulated. But at the same time, when we were treating with our treatment like the milk-fat membrane hydrolysate or the bacteria in combination with MFGM, so the markers had been suppressed to a large level, which was we could see that it was at the level at the baseline. And at the same time, the anti-inflammatory markers like IL-10 and TGF-beta, the expression of that increased to a huge fold, which shows that the treatment, the MFGM treatment and the MFGM LGG treatment was able to invoke a lot of these anti-inflammatory reactions. So now we wanted to see that this treatment of the bacteria that is treated with MFGM, when it is moving towards the acid and the bile in our system and all, whether there is any protective effect or not. So here we could see that when the bacteria was treated at different pH 2.5 and 2 or 3.5, as against the control, the MFGM was giving some protection, and the kind of reduction that we could see during exposure with acid, it was comparatively coming down. And in all of this treatment, the sequence was the same, that the effect was seen highest with the human MFGM and then goat and then the cow MFGM. Similarly, when the bacteria was being treated with bile at the level of 0.5 and 1.5%, in the control we could see that after 3 hours of exposure, there was a cell death that was happening. But when it was being treated with the MFGM, the cell death was comparatively much lesser. So we could see that there was so much of protective effect that this treatment of MFGM was giving to the bacteria. And this is one of the assays that we normally do in order to study the cell surface characteristics, which is also called the cell surface hydrophobicity, where we dip the bacteria in different hydrocarbons and we see that what is the change in the hydrophobicity. And in all of the 4 hydrocarbons that we studied, hexane, diethyl ether, xylene, and chloroform, here we could see that in the same sequence, that is human MFGM, goat MFGM, and the cow MFGM, the cell surface hydrophobicity was increasing in an increasing manner. Then these are also other parameters like co-aggregation and auto-aggregation, which also shows us that the bacterial cell surface, how good it is, how it is able to adhere to the intestinal epithelial cells, how it is able to colonize the intestinal epithelial cells. So at all the treatment levels, we saw that this human LGG, human treatment, goat treatment, and cow MFGM treatment, it was sequentially improving the cell surface properties, which is enabling the bacteria to adhere more. So which was kind of explaining that why we could see so much of improvement in the initial addition assay. So then we wanted to see whether the in vitro study can be replicated in terms of the in vivo study, and then the same kind of combination we fed to the animals. And these are the 9 groups that we had, and like, these are the different combinations. Where the first one was the negative control, second was the positive control, and these other was the treatment controls. So the treatment controls, first one was the cow MFGM only, goat MFGM only, human MFGM only, then the bacteria only, and then the combination of bacteria and the 3 MFGM. And after feeding the animals with these combination, after feeding for 28 days, we induced the colitis, the inflammatory bowel disease, by feeding DSS. So one of the very important marker of DSS colitis is weight loss. So here we could see the red mark that during DSS induction there was weight loss, because of which it was coming down, but at the same time those animals that were fed with different treatment, the reduction of weight was in a certain way limited. Feed and water intake is a marker of bad health in a way, but here we could not see much effect in terms of food and water intake. The disease activity index is kind of reflective of body weight, and also there are some parameters that is linked with stool and all. So here we could see that the disease activity index increased to a large extent in the disease, but at the same time when it was given the other kind of mix and all, the disease activity index was limited. The colon length, there is a shortening of colon length during colitis because of the inflammation and the shortening of the cells and all. So, and normally the colon of the mice is somewhat around like 8 to 9 mm, and during shortening it comes to around 6 mm, so 6 centimeters. So here we could see that there was shortening in the control, DSS control, but other treatment it shows comparatively better length in colon. The gut permeability, so the leaky gut issue that normally results in colitis. Here we could see that there was a lot of infiltration of the FITC that we fed to the animal, but at the same time the leaky gut that was normally results in DSS colitis, it was controlled to a large extent, and especially if you see in human and LGG treatment, it was coming to the baseline, so that it was resisting to a large extent. And myeloperoxidase is an enzyme that is found in neutrophils during inflammation. It percolates. So so which is also a direct marker of the inflammation that was happening. So here also we could see that the myeloperoxidase activity was greatly reduced in the treatment, with the highest in human plus LGG. So gene expression by RT-qPCR, tight junction proteins like occludin, claudin, and mucin. In all of these, we saw that the human plus LGG treatment was giving a very good response, and the tight junction proteins was highly expressed, and we could see that the barrier properties was kind of restored in the treatment. And at the same time, the pro-inflammatory markers like TNF-alpha, IL-beta, interferon gamma, and TLR2, so these also show the same trend, meaning that the inflammation was controlled to a large extent in all these treated animals. The anti-inflammatory markers that we studied by gene expression was IL-10, IL-4, and IL-5, and anti-inflammatory, just like the pro-inflammatory, it was showing a reverse trend. And in order to verify at the level of protein, we also run ELISA, and two principal cytokines, the IL-10 and TNF-alpha, showed the same trend that we would see in the gene expression studies. So an additional work that we also did was the fetal metagenomics. So fecal metagenomics in the healthy animals after treating with 28 days before the DSS colitis, we could see that the distribution of the microbiome was very uniform across all the species, across all the treatment, but as the DSS was induced, we could see that it was kind of disturbed, and there was aberrations in many of the microflora. And one of the very important marker that we study is firmicutes to bacterioids ratio. So from firmicutes to bacterioids ratio, we could get understanding that what is the level of disturbances or dysbiosis that's happening in gut microbiome. So in this DSS, which was given only DSS, the firmicutes to bacterioids ratio was coming to around 0.5. Firmicutes to bacterioids ratio in a normal microbiome is 1, almost around 1. So in DSS, we could see that it was coming at the level of 0.59. But when we look at the other treatment parameters, there was increase in the firmicutes. Obviously, we was feeding LGG, that is Lactobacillus rhamnosus, so there was increase in firmicutes also. But even when we are feeding only MFGM, so there also we could see it was helping to increase the firmicutes, which was giving a very good FNB ratio. So this was again to show that what are the bacteria that was very apparent in healthy, and then when the disease was induced. Yes, sir. So it shows that what are the different bacteria that was increasing and decreasing in different treatment animals. So these are again the different bacteria, the bacteria that are involved in short-chain fatty acid production. These are the major short-chain fatty acids that these bacteria produce, and what's happening in different groups that we studied. Furthermore, we also studied the fecal metabolomics. So fecal metabolomics, normally we studied the organic acids, short-chain fatty acids, and here it's just the group distribution. So when at the level of 28 days, it was a very uniform distribution, but during disease indication, the DSS group formed a separate group, but other groups were showing very close to the control. So that means even the metabolic fingerprint was pretty similar to the initial. So this was again the distribution of the short-chain fatty acids, the branched-chain fatty acids, so where it was increasing and when it was decreasing because of treatment. So just to look at the proteomics, we also carried out proteomics to look at, like, at the proteomic level what's happening. And in the bacteria, these were the top 10 proteins that was being highly expressed because of the MFGM treatment, and it was all involved in either the improved stress response, which we saw in terms of the improved acid tolerance, bile tolerance, or essential peptidoglycan synthesis. So that means the bacteria was becoming more strong because of MFGM treatment. And these are all other proteins that was down-regulated, which kind of showed that the physiology of bacteria, what was happening during MFGM treatment. And again, we try to identify what are the proteins that is present in the cell by running the proteomic against the human database. So many of the proteins from the MFGM, it was kind of maybe coating the bacteria, or maybe it was entering. So we did not exactly look at the cell surface proteome, but the overall proteome. So even though that we were washing the cells with buffer, but we could see that the treatment allowed many of the proteins to either penetrate, or it was forming a kind of coat in the bacteria that allowed the bacteria to show a different response in the in vitro, in vivo inflammatory studies. So in conclusion, I would like to tell that our findings demonstrate that species-specific milk-fat membrane can potential probiotic efficacy. So we normally study probiotic bacteria in a separate system, in a lone system, but in food system, when this bacteria are interacting with these kind of components, there is a lot of changes that's happening, which may be good in many ways, which may not be, which obviously requires, I think, studying. Obviously in MFGM we saw that depending on the species there was a lot of changes that is happening in the bacteria, and that offers a promising strategy for different, like, different kind of probiotic based functional food development. In infant nutrition also they are adding a lot of this MFGM nowadays, so depending on whatever the data that comes, so infant nutrition can also be like improvised in a better way, and therapeutic dietary intervention for intestinal disorders. Obviously further clinical models needs to be tested in order to see that whether it can be really reflected in the clinical levels. Thank you. Any questions?
Question actually, we will provide your contact number and email IDs to others. They will call you.
No, I think so they can ask me later also.
Yes,
Thank you.
So now our second speaker is Dr. Kaushik Kamri. He is a technologist and principal scientist at NDRI Karnal and well-known scientist in the field of indigenous dairy products, functional dairy products, etc. So I will invite sir to reach present.
So, respected dignitaries in the dais, ladies and gentlemen, and dear students, I am Dr. Kaushik Kamri. So, I will be presenting before you a research findings titled Prebiotic Potential and Antioxidant Properties of Raw and Ripened Banana Powder in Probiotic Drinking Yogurt, that is lassi. So, after a high research of Dr. Pradhan, so there is a down-to-earth research about lassi, okay, which we all mostly we release to drink. So all we know that the probiotics are basically live microorganisms that when administered in adequate amount, so confer health benefits to the host. So there are many health benefits which is suppression of cancer, improve gastrointestinal immunity, prevention of diabetes, improvement of gut health, etc. So there are certain foods which probiotics likes to eat. They are known as prebiotics. Okay. So mostly these prebiotics are mostly very costly ingredients like inulin, FOS, fructooligosaccharides, sometimes galactooligosaccharides, etc. These are all important molecules and they are very costly. So basically we wanted to know our powders manufactured from our good or abundantly available bananas whether can act as a prebiotic for growth of some of the probiotic microorganisms that are present in lassi or not, right? So that was the aim of the study. We first developed the product and then we wanted to know how long they remain drinkable when packed in different packaging materials. So why banana? So everybody wants to go banana, but it is because it's one of the largest agricultural commodity that is produced in India. So among all the agricultural commodities that is produced in India, banana holds the 9th position. And banana is one of the food which provides highest calorific value, around 400 calorie per 100 gram as compared to 1 to 2 gram per gram of calorie for most of the other food. And it is one of the cheapest source of various nutritional ingredients as well as energy to the human beings, right? So now the question is, if we convert banana into the powder, whether this powder could be also a good food for the probiotics or not. So there is no, another advantage is there is no safety study required because it's a conventional food ingredient which is used in lot of different types of foods like beverages, even it is used in cosmetics, pharmaceuticals and animal feed. But the main challenge is its solubility is very less soluble, and maybe whatever the product we are making, that will carry forward a banana flavor which may not be liked by everyone and which may interfere with the natural flavor profile of lassi. So you can see the probiotic is one of the product whose marketing is ever increasing. So that's why we take a probiotic product and lassi as a vector for carrying the probiotic microorganism. The microorganism we tried was Lactobacillus plantarum MTCC 5968. Right? So another thing is that these most of the probiotic, if you check at the nutritional content of the most of the probiotics that is available in the market, they does not contain any but this natural. Continue with the mic. So this natural banana, it also contains a high amount of fiber. So along with the prebiotics, the natural banana may be a mode of increasing the helpful fiber content in lassi, right? So the objective was to the standardization of processing parameter for production of banana powder fortified prebiotic lassi and study the shelf life of this developed product. This was a simple research work that we had carried out. So for that, we tried various changes in various processing parameters. The ingredient we started with was milk, which contains 3% fat, 9% SNF, and we added banana powder in various quantities into it. So we had tried 3 different types of banana powder. This raw banana powder, ripened banana powder, and mixed banana powder, and we tried their limit from 0.2, 0.5 and 1% level. And as we have told you that they are very less soluble in the milk system, so we tried high shear homogenization at 3 different levels, that is 6,000, 9,000, 12,000, to know how it's affecting the solubility of banana powder as well as how it's affecting its hydration of banana powder in milk. So then it was heated at its running lassi and then inoculated. The providing microorganism was at the rate of 5%, and then it was incubated for 37 degrees centigrade at 37 degrees centigrade for 4 to 5 hours, and then to prevent thawing off, we tried high methoxyfloxacin at 3 different levels, and to improve the solubility and suppress the banana powder, so we also tried external addition of some flavoring ingredients. We tried close spectrum, right? So here basically we have seen the composition of different types of banana powder that we have tried. So if you look at this slide, you can see, so this contains the high amount of resistant starch, so 47% for raw banana powder, and which was very low in case of ripened banana powder, and around 44.5% in mixed banana powder. Okay, and almost 36 to 40% of the resistant starch was digestible. That means more than half of these were indigestible, and which food, this indigestible starch portion may act as a prebiotic ingredient for the probiotic microorganism that we have tried, right? So we can, as you can see here, so as we increase the speed of the high speed homogenization, the solubility of the banana powder was increased, and the swelling power of the banana powder was also increased as the homogenization speed was increased from 9,000 to 12,000 RPMs, basically helping us to solubilize the banana powder in the milk system as well as improving the mouthfeel profile of the powder, of the final product. Okay. And then as we increase the high methoxyfloxacin content, you can see here, so the weighing up was being reduced, and in case of, you can see addition of the raw banana powder, the effect of weighing up, prevention of weighing up was better as compared to other two variants of banana powder, that is ripened banana powder or the mixed banana powder, right? So the mixed banana powder is basically a mixture of ripened banana powder and the raw banana powder. And as we are increasing the addition rate of raw banana powder as well as the pectin, we wanted to know how its sensorial attributes has been affected, if the prepared lassi was being affected. We can see here, so addition of 0.5% of raw banana powder and 0.15% of pectin is basically provided a combination of lassi which is highest, which was most liked by the panel of our judges. So we selected these as a formulation in our further studies, right? So you can see, so when we added rose flavor at the rate of 0.05%, so the overall acceptability was slightly improved. Okay, so the banana powder contains some amount of phenolics also, which is antioxidant activity. As you can see here, as you increase the banana powder content, so the radical scavenging activity of the lassi was increased, and it was more in case of mixed banana powder. Right? So as we increase the banana powder content, so the phenolic content was increased, so which was basically increased the DPPH radical scavenging activity of the final product. Right? So what is the effect of raw banana powder on the probiotic count? So we have tried to use this strain which was used to prepare the lassi as well as with the probiotic strains also. So we can see, so as the quantity of the raw of the banana powder was increased, the probiotic count was increasing slightly, and it was maximum when raw banana powder was added at the rate of 1% and mixed banana powder was, ripened banana powder was also added at the rate of 1% in different samples. But these samples we did not take because these samples has a marginally less sensorial attribute as compared to the banana powder, in which the banana powder was added at the rate of 0.5%. Okay. So this is basically a approximate composition of the product that we have finally standardized. So you can see here, so it contains digestive resistance of 0.19%, and the dietary fiber was also, some amount of dietary fiber also present in the final lassi, which is around 0.54%, and our control sample does not have any dietary fiber. So basically this, even the small amount of digestible starch, when they are coming from the raw banana powders, so they have a profound effect on the growth of the probiotic microorganisms that are used for making the lassi, right? Then is the next step, we wanted to know how long they feel good, and we packed the product in 3 different packaging materials: glass bottles, poly pouches, and polyethylene external bottles. I can see here, so as the storage stability was increased, the acidity was increased across the packaging material, but the increase was most in case of samples which are packed in LDP pouch, and the least was in glass bottles. Okay. And the weighing was also increasing. The glass bottles has a less study effect in preventing this weighing off at least up to 12 days of the storage, but as the storage was increased from 12 to 15 days, this weighing off was suddenly increased. Right? So the radical scavenging activity, as we stored the product, we got increased, and in case of the raw banana powder added samples, so this was more. Okay, the phenolic content was basically almost the same throughout the storage period, and it will reduce as the storage period increased from 15 days. Probiotic count was for a continuous increase, but the probiotic count was the extent of reduction was less in case of total samples, so whether it is packed or glass bottles, right? So the viscosity was being reduced because of the chloride degradation of some larger molecules, right? And the product was acceptable up to 12 days. 12 days was the safe life of the product that was developed, and it was safe for the control sample also, right? So in conclusion, so we can see the addition of raw banana powder among the key persons of the powders we tried has demonstrated prebiotic potential and better antioxidant properties to lassi as compared with control sample. Okay, and the base homogenization as well as solubility, the effect was provided by the speed of 19,000 RPM, and the sample which contains 0.5% of the powder and 1.5% raw shelf life of 12 days. So which was around 2 days more than the normal lassi which is available in the market. Right? Thank you.
Now we will provide you the contact details, so anyone if interested they can come. So next our speaker is Dr. Kritika Avenkar. So Dr. Kritika Avenkar is an assistant professor in the School of Science at now Channa University, Vadodara, Gujarat. Now I will invite her to speak her topic. And I will request to
সবাই সামনে চলে এসো তো
লাইট চলছে না, সবাই সামনে চলে এসো। তাহলে পিছনদের গলা ধরে যাবে। Spike is not working, so they must come forward.
চলে এসো
अरे सब बंद करा रहेगा, आ जाएगा, ऐसे बोल लेंगे। ओके
सभी लोग लेते हैं, इसकी प्रॉब्लम नहीं
এই বন্ধ করো আওয়াজটা বন্ধ করো
silence.
संजीव कुमार नहीं है,
दो लोग नहीं।
So, good evening everyone. I am Dr. Kritika Avenkar from Nagarjuna University, Gujarat, and I shall be presenting my work on fermented legumes as functional foods linking solid state fermentation bioprocessing technique to the anti-diabetic activity. So the question is, why anti-diabetic, right? Next. So if we look at the global diabetes challenge, if we look into the prediction values, by 2040, 642 million people are prone or they are the ones who are going to have diabetes. However, if we look into the Indian scenario or Southeastern Indian scenario, wherein 140 million people are supposed to be or predisposed to be diabetic. Wherein, next please. Next please.
Next.
you have that on.
Next please.
Next, below cell.
Do this.
Below, नीचे रखा हुआ है.
This one.
Below button.
So if we look into the Southeast Asian part, what we can see is, looking at the Indian scenario over here, there is a huge discrepancy in terms of an age by prevalence of diabetes, which means even an individual of 20 years to an individual of 79 years is going to have a trouble with that of diabetic condition. Henceforth, there are certain highlights for the diabetes. We shall move ahead because we know that how serious diabetes would be. It's a chronic metabolic condition which is going to have an impaired insulin or impaired secretion of insulin, ultimately is going to disrupt the blood glucose level or the blood glucose metabolism level. Henceforth, what we are going to try is, or what were our hypothesis for the present work was, we can see over here that whatever we can say a polysaccharide rich diet which we are consuming or the carbohydrate diet which we are consuming, it undergoes some series of hydrolysis wherein the starch is going to be converted into maltose with the help of an enzyme which is called as the alpha amylase. Now from where is this alpha amylase coming? It is coming from the pancreatic juice or the pancreatic acidics. This alpha amylase further we can see breaks down into the maltose, and this maltose is further converted into the glucose with the help of another enzyme which is coming from the small epithelial cells which is called as an alpha-glucosidase. So we wanted to test if by default, if I am able to use some legume-based conditions or processing my legumes, are they capable enough to give rise to certainly something like a bioactive compounds? Are these bioactive compounds also rich in terms of potential levels? And can they also act to reduce this alpha amylase activity? This is what we wanted to check with the help of a positive control, and the positive control which we have used for the present study is acarbose. Acarbose is a normal drug which is used for the anti-diabetic therapies. Moving ahead, as I said, legume is the one which we have considered. So 3 legumes were used for the study. Now why these 3 legumes? We can see the first one was labla bean, also known as an Indian bean, which is commonly in Gujarat called as an wal or wal papri. And we have seen that it is widely used in the other parts of the country as well. It's been cooked well, culinarily also important, reported to have a phenolic content and also reported to have antioxidant power. Moving on to the next one, what we have is a cluster bean, which is again culinarily staple. However, the pods are being consumed, not the, we can say that not the seeds which are being consumed. Followed by that, another significant values which we have for is for polyphenol and the GABA. GABA is by default acting like a vasodilator, so in turn, it will also help somewhere in the vicious cycle of anti-diabetes as well as that of anti-hypertensive therapy. The last one what we have is an Albizia bean, and it is used as an animal feed application, not being used as such for the human consumption. However, being drought tolerant, grown abundantly in Gujarat and also reported for a bioactive source. All the 3 legumes which have been considered for this study because of their underutilization. So that is why the 3 legumes were being selected. Moving ahead with the research flow, if we can see over here, I have divided my presentation as well in the 3 different stages, where the first phase is about metabolomics, wherein the first stage of metabolomics is associated with that of germination. So the legumes which I have considered, I have passed them through the first level of, we can say, bioprocessing, and the most simplest way of bioprocessing is germination. So germination basically helps to reduce the anti-nutritive factors such as in terms of phytates and tannins. So that is where we have germinated legumes. So we have prepared a sample followed by the LCMS was done, and then taking account of the huge classes, subclasses of all the different metabolites. Moving ahead, we looked into the solid state fermentation, which was the actual way, or we can say a second bioprocessing technique which we have used. The whole idea is, can this fermentation using a particular strain of a fungi can elevate the metabolites or not? So that was our second concept. Moving on to the third one, if enrichment is there, is it capable enough to lead to an inhibition in terms of alpha amylase? So alpha amylase particularly because if it will reduce the amount of maltose, it will try to work upon the substrate as such directly. So in order to consider the first type, what we have done is we have looked into certain parameters where we can see protein values, phenol values, and ascorbic. Also some antioxidant parameters for the germinated beans were carried out. Wherein we can see, yes, all 3 of them were reported to exhibit the good levels of polyphenols. Also in terms of antioxidant activity, they were able to get some good results. But the question was, is there reporting any kind of alpha amylase inhibition or not? Now this analysis which was being carried out, it was for the aqueous samples. So naturally we can see the activity which is reported instead of microgram, it is reported to be in milligram. So the acarbose will show an activity in microgram, while our legumes right now showed it in somewhere in the middle, right? Henceforth, moving ahead to our phase 1, where this germinated legumes, as they are capable enough to show some of the certain good activities, what was the metabolites which were reported from them? So we have gone through this positive and negative mode of spectra which were developed. Followed by this was the we can say this was the distribution of the metabolites. Lot of phytochemicals were reported in each group, and again they were not reported to be similar also. In some cases it was alkaloid which was more in consistency, somewhere it was flavonoid which was more, here it is less, and so on. Also, interesting thing was we were also able to report certain kind of peptides and dipeptides just after germination as well. And this peptides could again help in as an act as an anti-diabetic activity. Moving ahead after the metabolomics profile, we can see over here that there were some compounds which were tried to be overlapped between the three groups. And if we try to spare those common compounds, there were certainly some unique compounds which were there. Wherein cluster B was the highest category of 81 compounds. There was a heat map which was developed based upon the score of 50 and abundance in the germinated seeds. And then we have seen, after looking at this particular heat map, we can see that one or two compounds, something like in cynarocyte, another one which was over here miraxanthin, they are previously reported for their in silico activity, where they are already reported to be anti-diabetic or alpha amylase inhibitory activity, which somewhere tells us that okay, we were in the correct path of metabolomics. And however, now we wanted to check into the terms of what is happening in the solid state fermentation. We can see over here, Aspergillus avamori colony was grown on the PDA, followed by that the colonies were, the samples were isolated, they were stained using lactophenol blue. This was the basic methodology which was adopted, wherein the legume substrates were there, and followed by this, the sampling was done for 0 to 120 hours. 0 was considered to be controlled, 120 hours was almost a period of 5 days. Where the main objective of this part was, is there any kind of phenolic liberation or not? So this were the different samples which were collected at different periodic times. And then quickly if we see, this is basically for lab germinated, that was the first seed. And henceforth we can see at a period of 96 and 120 hours, the protein levels are comparatively higher as that of the control group. Likewise, phenol also, DPPH as well, ABTS. PrAP as such, we can see over here a slight major change was been reported. However, no major difference in terms of alpha amylase was reported. So we wanted to then look into what will be the enrichment specifically by isolating or extracting the phenolic compounds which were there. Likewise, we can see for the cluster B protein here, one of the interesting thing was that there was no much more amount of change in terms of alpha amylase also in the PrAP, which was categorically seen in case of the lab. Moving ahead with that of the last being, that was Albizia. Herein we can see again alpha amylase was reporting a similar type of behavior compared to that of your cluster B. So henceforth we wanted to move ahead and see what are the changes or what are the post-fermentation dynamics which are taking place. And then, as our objective was for the phenolic study, we have done a phenolic extraction and then we have used it, separated it, quantified it using HPLC. And then using the standard graphs, we have tried to find out which are those particular phenolic compounds which are going to be present. So, if we look into this, we can see some of them were having more amount of gallic acid while some of them were reported very low amount of gallic acid. However, most of them were seen to be reported in terms of ceramic, ascorbic, ferulic, catechol and so on in all the in case of vanillin and quercetin, it was only the cluster B samples which have reported, while lab and Aspergillus, lab and Albizia, they have not reported any kind of vanillin. The last one is the alpha amylase, wherein the same phenolic extracts were then looked into the IC50 value in terms of an alpha amylase inhibition, where Albizia, Saman has reported to be having a lowest IC50, which means it has a stronger inhibitory potential even compared to that of the acarix. Which means Albizia, which is categorically underutilized for a human-based condition, has to be further validated, has to be further studied as well. To conclude, I would say that solid state fermentation using this Aspergillus avamori has actually amplified the bioactive potentials in terms of phenols. Metabolomic signatures are also correlating with that of alpha amylase, where particularly miraxanthin and amylicin and cynarocyte is going to be playing a role over here. Lastly, fermented legumes are showing its activity, contributory activity, or we can say almost nearby activity to that of an acarus. And the last one is functional food platform for the diabetes prevention and management, where all these three legumes could be studied further, could be evaluated and validated further using in silico studies and also in vivo studies. So docking is the first line of work which we plan to do across this part. Followed by that, we plan to go across this bioavailability studies using cathode cell lines. And the last is not only the bioactive compounds in terms of phenol, but also the bioactive peptides is what will be isolated and characterized. These are some of the references which were used. These are some of the publications as a part of initial study from metabonomics of labnan and clostridium, which has been done in my lab. And this work was of my student, that is Komal, along with my 2 MSc students who have worked with me for this. I want to acknowledge the Department of SEP IIT Mumbai, where we have carried out this metabolomics analysis. I would like to thank my parent university Navratna for giving an infrastructure and support for conduction and even presentation of this. Thank you.
Now I will request to next speaker, Mr. Mandal, and I will request Mr. Mandal sir kindly fix your panel within 10 minutes.
वो पॉइंटर दे दो सबको।
So, very good afternoon. Professor Vishnu, my good friend, my close friend, on the dais and off the dais, technical scientist. Actually, I am summary of the school board. Therefore, I will present my research, what I am doing for last more than 12 years in the field of low alcoholic beverage. Low alcoholic beverage, and before that, I offer my tribute to Professor Jyoti Prakash Kamal, the member of SASNET, as well as he actually pushed me to initiate research in this work. So, why it is so important till now? This is a good, very important question, and because this sort of drink will keep consuming for more than thousands of years. Now, Government of India given lot of emphasis vocal for local. Okay. Therefore, as an expert member of the FASI, I am also an expert member of FASI in the field of alcoholic beverage. Very recently, last July 2025, Government of India gazetted the local beverage, 54 local beverage. Why? And because this will expand huge market. And because till now Indian people depend upon the foreign liquor. Okay. But we have enriched beverage. And these are consuming for more than 1,000 years. And it having the cultural relation, cultural importance. So, this is the notification Government of India Gazette. Therefore, anyone, today I will discuss, that portion is mentioned, highlighted, which is locally called in the part of the Bengal and other part of the country as the Handia or Hania. This is a low alcoholic rice-based fermented beverage, and this sort of beverage is very popular in North India and the Himalayan zone. So basically, alcoholic beverage can be divided into two kinds. One is the cereal-based, another is non-cereal-based. Cereal-based means these are mostly prepared from rice, beside millet, barley also substrate, and these are the non-cereal group of beverage. So what point we have to think, and because in case of non-cereal-based, these are mostly naturally fermented. Therefore, we are unable to control them. Naturally fermented beverage, therefore, we are unable to control them. But we could standardize the starter-based alcoholic beverage. We could optimize the starter-based alcoholic beverage. And what I stated that I am working more than 12 years in this field. So basically this is a rice fermented beverage. It has very extensively used. Many is no botanical importance. Tribal people, what today one speaker told that this is useful for the gastrointestinal health. It is well established. Tribal people, when even small baby suffering by the gastric problem, they provide this sort of drink. This is a simple preparation. They taken the rice, thereafter added the starter, and then ferment within the oven pot, and then diluted, and then consuming. This is also now, as per the FASI, it is now as the ready to drink alcoholic beverage. Ready to drink alcoholic beverage. Ready to drink alcoholic beverage. This is what I stated, that this is very popular. Just you see Red Star all over this central India. It is popular. Particularly in the West Bengal, there are many government shops. And after addition of the starter, this starter is very important. These are not the old ferment as like the dohi or other thing. These are not old ferment or isolated organism. These are basically plant-based organism, which are we designated as the endophytic organism. How starter ball is made? Mostly tribal people selected some specific group of herbs, thereafter collect their rhizome and make a ball using the rice dust. This is the actually starter tablet. And this sort of practice remained very popular in the Himalayan region, in the central part of the India. And the microbes which remain within the plant, that actually play active during the fermentation. Therefore, these microbes are called wild microbes. Wild microbes means previously they are not exposed to environment. Therefore, there is no chance of the mutation. In case of normal starter, what thing happened? Organism like the COVID, they can be changed naturally, change their behavior in case of starter bacteria. But these are wild, therefore there is no chance of the mutation or alteration. So this is the textural change of the rice then. Oh. A huge number of microbes are there. If you see, this is a very interesting interesting characteristics of fermentation. This is basically what they made. They first boil the rice, thereafter sun dried, and thereafter mixed with the starter tablet. Sun dried, thereafter mixed with the starter tablet. So fermentation basically one sort of solid state fermentation. And the fermentation process is multiphasic. That means whenever that initiate, that catalyzed by the mold, amylolytic mold. Therefore many people highlighted this tablet as the amylolytic starter. And after 2 days, whenever the amylolytic mold secrete amylase, they decompose the starch and create an anaerobic condition. Then only lactic acid bacteria and yeast abides. And whenever lactic acid bacteria abides, thereafter you can't find any fungal residue. And lactic acid bacteria having the catalytic ability to destroy all the fungal residue. So ultimately you will get lactic acid bacteria and the yeast. This is the final product, not any molds. Oh, this is I'm just showing one point is that this is the DGG gradient electrophoresis analysis, where we just highlighted that there are 6 types of lactobacillus. There are 6 types of lactobacillus. And during the fermentation, we have analyzed the fermented material contain around 160 number of reactive metabolite. And we have linked with the participating microbes. We have linked that 29 metabolites are directly synthesized by the participating microbes. So next question arises, from where remaining metabolites are come? These are basically plant metabolites. During catalysis, they are separated, therefore enrich the material. So this is the link, and during the course of fermentation, pH gradually decline like any fermentation, And interestingly, during the course of fermentation, just you see the content of G3, G4. This is the different, this is the oligosaccharide. G1 is the monosaccharide. But most interesting point is that there are some oligosaccharide also deposited in the fermented material, which are very much beneficial, as many speakers already mentioned. And many volatile compound synthesized. And this is a free amino acid, that is mostly, it has identified that yeast is the major so-called source of the amino acid. And during the course, hydrate degrade, this is a normal course, and mineral quantity enriched, as because normally in the cereal, mineral are entrapped with the phytic acid. When phytic acid degraded, then mineral become free, or that is called bioavailable. Rice, within the rice, if you analyze the atomic absorption spectrophotometry, you will see that there is mineral present, but these are not bioavailable. Okay, but during fermentation they become bioavailable. This is the alcohol, we have analyzed it extensively. Alcohol content and vitamins, B type of vitamins, ascorbic acid, also fatty acid. Different types of fatty acid. And most importantly, essential fatty acid also increase course of fermentation. Phenolics, we have extensively examined the phenolic profile. Around 6 to 7 phenolics are enriched. This is the major point of difference between dairy-based fermented product and cereal-based fermented product. In case of dairy-based fermentation product, this is devoid of phenolic compound. But in case of cereal fermentation, you will get enough number of quantity of phenolic which having the antioxidant activity. This is the in vitro, and this is the macrophage cell line, where we have identified that these phenolics induce the activity expression of NRF2, thereby increase the total catalase, peroxidase, these group of enzymes. Apart from this, it having the antimicrobial activity, particularly active against the different species of Shigella. We have examined highly active. Different antimicrobial compounds are synthesized. Different free radicals are synthesized in the fermented product. Not only the peptide, other material also. And we have studied its anti-obesity effect. And this, it is well established that in case of probiotic research, multi-species, multi-strain probiotics are effective. Therefore, in the market, whatever probiotics are available, most of them are the multi-species and multi-strain probiotic. And the composition is contain different group of healthy bacteria, and it can modulate the total lipid metabolism. Total lipid metabolism means it control, I am not going into the details there because sir told me I have to complete. It changes the total lipid metabolic pathway, therefore obesity decline. So these are the expression. So my hypothesis is that these fermented products contain fatty acid, phenolics, probiotic, oligosaccharide and other compounds which modulate the activity of gut microbiome. And that has been, and this whenever gut microbiome activity active, then synthesize till day few intermediate have been identified. One of the group is the short chain fatty acid. And we have also analyzed, but there are many intermediate may be present. We have analyzed the short chain profile activity, their quantity acetate, propionate, butyrate, their quantity increase. And so in conclusion, this sort of what I stated earlier, that this is the new era of so-called, particularly for the startup. This is a good scope to initiate this sort of business. Just you see that these are rice fermented, okay, very your setup, no need huge money, but as government mention its standard, therefore enough scope to exploit. So there are already PSCI approved. I am not going into the details. So sir, thank you. Yes sir, बताइए.
At the end,
Okay, just one question.
हाँ.
In that
presenting low alcohol beverages, but never you have said how much low means what.
Sorry. That is table 4 of the FASI, this regulation, where is clearly stated what is low alcoholic. Till it is now 6 to 7% is now considered as the low alcoholic beverage.
7%?
This is very low.
Because of that you have not disclosed in detail.
No, already remain in the website. Details, FASI regulation with all details.
Okay, thank you. So now, last but not the least, next speaker is Dr. Narendra Raju, and he is well-known food scientist, dairy technologist from NDRI Karnal. So now I will invite to present his
Very good evening to one and all, respected chairman. At the outset, I thank the organizers, especially the coordinator Susan Aspect, as well as the organizing secretaries of this conference, for giving me a chance to deliver this talk, as well as very intelligently keeping this particular 2 sessions, which was preceded by Professor Mandal, and this session on wine at the fag end of the day. So thank you very much for keeping this at the fag end of the day, and it's the right time to hear something about the wine here. So I'm here talking about the biocircular valorization of whey for sustainable wine production. As he rightly mentioned, there is a need to develop indigenous kinds of solutions for our dairy industry problems. So in this session, let us just quickly look at what is the current whey production in the world, in the country as well as in the world. So we know that there is a sustained problem of utilization of whey by the dairy industry. Although efforts have been made, still whey is a large amount of byproduct being generated by the dairy industry. Now there are different types of wheys. I am currently focusing only on the paneer whey, how to utilize it at industrial scale, but for the information of the house, those who are not aware, we have acidified wheys such as the chakka whey or the cheese whey or mineral acid acidified wheys, and also the paneer wheys. So we know that the composition of these wheys also differ, and the current context is related to the utilization of paneer whey, and we know that the protein content of this whey is very low compared to the other wheys. But the utilization issue of any type of the whey is the same, so there is a requirement to address this issue at a global level. If you look at what is the problem, it contains very large amounts of biological oxygen demand, and so it has to be utilized. It can be utilized by several microorganisms causing different problems. So there are issues when you treat the whey properly in the dairy industry and pass it on to the municipal supplies or utilize it in the dairy industry, the environmental pollutions are minimized. So globally and historically, it has been utilized to convert or use this whey for spraying of the farm fields, maybe directly discharging into the sewage streams, or maybe as animal feed. But if you look at the progress that has been made in the utilization of whey since 1950s, there was initial utilization problems, but later they understood what can be done and then started recovering the whey proteins. So this era was considered to be as one of the golden era for the dairy industry, because very important nutrients were isolated from whey. But the issue of utilization of the protein devoid whey is still haunting the industry to recover the lactose and use the other liquid. But it is a cost affair to isolate the lactose and the proteins. And with the increasing milk production in the country, as in the morning also Dr. Tata, who is the Vice Chancellor of this university, also mentioned that the milk production is increasing, but there is no effective price being paid to the farmer. So a need for value addition is important. So it is expected that by 2050, 400 million tons of milk is being produced. So we need to look at what are the major ways of utilizing this milk and the value addition that can be promoted. So on and so forth. The consumption of paneer, which is considered to be a pan-India product now, is also increasing. People are now worried about how to utilize this whey that is being generated. It can be very effectively managed by treating the whey or by converting it into fermented product like wine. So this session will focus on how it has been used, and we know that there are different types of whey-based products. We can recover the proteins from it in the form of either concentrates or isolates. We can also convert them into fermented or non-fermented kind of beverages. And among the alcoholic beverages, there have been reports where the whey beer has been produced, as well as a number of whey wines. But non-alcoholic beverages are also popular, and some of the non-alcoholic whey-based beverages are already in the market as being produced and marketed by the startups. Now, it is not new that whey-based wines are prepared across the globe, especially in the Scandinavian countries. You also find them in the New Zealand as well as in Germany. In India also, a couple of decades back, work has been initiated and successful products have been developed like Acido Whey as well as Whey at NDRI in very particularly. Globally, if you look at the problems with whey and utilization for the conversion into wine, one of the earliest report in 1975 itself has identified the problem of utilizing in the 6th proceedings of the Food Processing Industries Conference held at the Chicago. Now, and a simultaneous concurrent event at Wisconsin in Madison. So these two, the conferences have highlighted that there is a need for utilization of whey for conversion into wine, but there are also associated problems like the way wine that is being generated is bland in taste. So they wanted to have different kinds of value addition in it by incorporating some artificial flavors like fruit-based flavors or natural ones. So the issue was very clear, and the issue is still haunting the industry because in the recent times, people have started looking at not only the utilization of they started understanding how to convert this protein utilized whey source for conversion into alcoholic beverages. One of the recent publications from the European country has again highlighted that there is still the problem has not been properly addressed. So this was, next please. So this was again looked at what could be the possible avenues to utilize this whey to convert into wines. So a number of fruits which have already been popular in the market can be incorporated, because in Indian context also, as Professor Mandal just mentioned, the need for the local wines is increasing because the imported wines tax or the duty is very high, and according to the recent report by the Ministry of Food Processing Industries, the wine market is booming a lot. Now, there are several benefits of consumption of wine, but when it is consumed in a moderate amount. So I'm not here to go into details of the health benefits of wine. Mostly the grape wine is very popular, but there are a good number of fruit-based wines. The process of the flow diagram for the conversion of whey into alcoholic beverage is very simple. This is for the grape wine production. But if you intervene with the incorporation of the must preparation stage by including the whey at that particular stage, and also instead of using Saccharomyces cerevisiae as a yeast species, because lactose is not being utilized by this yeast, so we need to have an intervention where the dairy yeast can be incorporated. So probably a blended yeast is needed. So this was the intervention that we made, and then try to address what kind of value-added product can be obtained out of it. And fortunately, there is a provision in the FSSAI regulations also that fruit wine, which is other than the grape wine, can also be produced with a minimum alcoholic content of 7, which is just a moderate amount of the alcohol what Professor Mandal just mentioned. Now, we have carried out work exclusively by utilizing the paneer whey of the dairy industry, and also a whey that is being considered at the northernmost part of the country, that is Ladakh, where we are currently working on utilizing the early fallen fruits of apricot due to the climate change that is happening. So the need for early fallen, or what we call the wild kind of apricots, is a major concern, so that the farmers of the Ladakh region can get some benefit out of the early fallen fruits. So we try to blend this by addressing the dual valorization, or by tackling this issue towards dual valorization. So apricot, worldwide, Turkey is the largest producer, but in India, the Ladakh is the largest producer, and whatever the Indian apricots are being produced, it is from that or the Trans-Himalayan belt. So we intervened by utilizing the whey that is deproteinated for conversion into musk, and the cultures of the yeast, wherein a blended culture was used, and we try to have a blend of this whey as well as the apricot pulp to produce a product which can be attractive enough, sensorily acceptable, and having an alcoholic content of at least 10 to 11%. So the standardized process is shown here, but due to limitation of time, I'm just skipping it. If someone is interested, I'll be able to talk with them more. So you can see here the percentage of alcohol that is being generated is not less than 12% by having a different kind of blend of whey as well as the apricot pulp, meeting the FSSAI standards. Look at the content of methanol here. It is very less or below the detectable levels, indicating that it is really safe, which is a problem with the countrywide, the country liquors that are being produced. So the other benefits include that a huge amount of antioxidant production due to incorporation of this, as well as the in situ fermentation. Now, the benefits of this dual valorization is that we are trying to address the problem of early fallen fruits in the Himalayan belt, as well as look at what are the best ways of utilizing the paneer whey, which is, the paneer production is booming, so there is no organized collection of whey as of now, which is a challenge, but yes, there is an option or the valorization of what we call opportunity. So there are several other issues need to be addressed before we actually promote this particular product in the market. Like, for example, we need to have a nutritional analysis in detail one, the one which Professor Mandal just mentioned about the phenolic content in detail, the vitamin analysis in detail, which are still going on at our lab. So with this, we'll be able to tell what are the major benefits of utilizing these two important commodities. So there are different pros and cons of the process that we use and the product that we have developed. But to conclude, we have addressed intelligently by utilizing two different raw materials which are considered as byproducts from the dairy industry as well as the horticulture industry. So this is trying to, we try to mitigate the problem of environmental pollution at both these industry levels. So with this, we could develop a sustainable solution to both of these industries, but to bring it to a market, like the founder of Red Cows, in the morning he mentioned, Narayan Majumdarji, that there is a need for value addition, and this product with detailed information could be one in the coming future. So thank you very much for your kind patience.
Sir, he has a question.
Any question, please? Yeah, yeah.
Yes.
Please, please.
Sir, the whey wine, which is now coming into the minds of the people, will it be a safer option compared to the regular wines that in the industry? And should we shift from that to this kind of product because of the health benefit?
Yeah, usually when the word wine comes to the mind, we always refer to grape wine. It has been well studied across the world, but these kind of blended ways need to be addressed in a scientific manner. So the scientific studies are in progress. So you just need to wait for a year or so from now, with which we'll be able to provide you more details about the nutritional benefits. So as a consumer, you can choose which wine is better for you, and then you can pick it from the shelf. So that is what I can say at this point of time.
Thank you, sir.
Thank you. Thank you.
Thank you, Dr. Ravi. So I congratulate all the 5 speakers. They have presented well on their topics.
We now come to a special and meaningful moment of this technical session. On behalf of the organization of OOPS StateCon 2025, it is our proud privilege to express our sincere gratitude to the esteemed chairman and rapporteur of this technical session 3. Now I request Dr. Subroto Hathi to felicitate our chairman sir and rapporteur sir. Thank you, sir, for your inspiring presence and valuable contribution. May I now request our chairman, sir, to felicitate our invited speakers of this technical session 3. Please come one by one, technical session. First, Divas Pradhan, Dr. Divas Pradhan. Divas, Dr. Kaushik Khamrui, Dr. Krutika Abhayankar, Dr. Keshav Chandra Bondal, Dr. P.N. Raju, and lastly, Dr. P.N. Raju, and then one group photo with the speakers. Godapi sir and Hathi, please join. Fukuda sir, please also you join. All the speakers, please join.
লাইটটা জ্বালিয়ে দাও না, লাইটটা জ্বালিয়ে দাও, অন্ধকার আছে। কাম
This side.
This side.
This side.
ও স্যার হ্যালো, এক্সকিউজ মি, একটু পিছিয়ে আসো না, সবাই পাবো। ভাই একটু পিছিয়ে আসো, সবাই পাবো। একটু পিছিয়ে আসো কাকা।
Thank you all for your attentive listening. Now we will be at the ground floor for cultural events as well as dinner. Thank you. So we will join here tomorrow sharp at 10 a.m.